Let me, okay. Yeah. Can you see my screen? Let me just to see here. Um, what do you are planning? Should I wait for Tina just a few minutes, or could we start it and when she comes, she present? Okay, I'm kind of lost here.
Hello, Melissa.
Hi.
Are you able to control the screen?
Yes, yes, but I was asking about if we should wait for Tina, or I should start.
Oh, please start.
Okay, okay, thank you. Okay, um, I will present some results. Instead, I put the information about the Holy Project here, the title, but I'm trying to resume here. Our project is, as all of you know, was very, very good explained by Dr. Abad, and then Caleb, and then the other colleagues as Vincent. Um, so we are working together with 12 countries, and then we are 14 institutions, and all of us working together with in long-term experiments data. So information. So we are planning to access information about the how this how time could uh, impact in carbon soil persistence. So this is very, very interesting when we have so many long-term experiments and so many long-term data sets, and then together with the modern tools. So let me try to here. Yeah, here are the institutions, of course. And then here, just a few tips again for our colleagues. So each researcher will present a concise update of the activities or tasks in each country, and focusing on major milestones and challenges, key findings if we already have, next steps for collaboration. Uh, why I'm speaking if we have key findings, because I think the first main step is organize and harmonize data set in the repositories. This is very um, even is we already have the data. This is very, very hard to do because we have lots of data. So this is a huge, not a big, but huge challenge. Uh, Here I put another information, kind of a list that I was thinking uh, to follow. So starting with me, and then from France, Hungary, Estonia, South Africa, Ghana, New Zealand, Norway, Germany, Uruguay, Canada, and a close discussion. I don't know if all the colleagues will start with us today, but yeah, I will. We will see. Um, from the Brazilian side, I think the main challenge is obtain the information or help to colleagues to put information in repositories. We started this project with 40 long-term experiments, uh, and together from the Brazilian side and the foreign colleagues. For the Brazilian side, almost I think all of us, all the experiments are from Embrapa. So as employees, we must to put our data in Embrapa's repository called HEADUP. And uh, from the colleagues from abroad, we are using Zenodo repository. So we agreed that we will have the two repositories having a copy, like working as a mirror each other. So we started from with Zenodo. We already have 10 data sets. Just this is just from abroad. The colleagues from Canada, France, Ghana, South Africa, let me see, Estonia, Hungary. I think it's just New Zealand. I think from abroad Brazil. We are just in waiting for Uruguay data set. They are having some concerns, but I think soon they will put the data there. And then from Brazilian side, we already have 11 data sets. It's different yet. So we have 21 data sets. It's just half of we started thinking that we will have, but I think as a data set and from historical for long-term experiment, this is very, very it is a challenge because the researchers already have the data, but they have no time to proceed this. This is hard because the researchers, almost the researchers has the data in her or your own computer, personal computer. So I think I'm very happy to have 21 data sets in for the colleagues' work. I'm sorry. Here is in a blank because, as David told for you yesterday, we are just starting because before this team start to work, we have to have the data set at least a good, I forgot the word in English, I'm sorry, a good union of informations from different sites. We have to have this in the repositories, and then we need to harmonize this because um, each country has their own methodology. And then, because of this, at the beginning of the project, we agreed that we would uh, do, or we would proceed at least one soil sampling, and then we use the same protocol. This protocol is we already have. This is um, in being revised to under review to be published. And then we have protocol for methodologies and protocols for data use. So both are under review. And then let me see. Another thing we are as a information for the how we are proceeding. Another thinking we are together in the project is about the podcasts in Work Packages 3 and social media that Caleb presented yesterday. And then this is just um, a script about the structure of our podcasts. And then I hope at the end of this workshop, we have a list with the colleagues who would like to present, because at the beginning we were planning that we would be very, very good if we produce at least one podcast per institution, and then organizing and these activities. So as you can see, it's not a big podcast. We could have a podcast of until 10 minutes, 9, 8 minutes. I think if 4 podcasts is it's a good information. And then for presentations, a huge time, especially in the nowadays. The people likes the information come very fast. Otherwise, the people, how do I say, accelerate the voice. So we need to be um, very concise and objective. And we are thinking how to proceed this for this, especially for this new generation. So I hope to have help from the younger researchers because I think the people, these colleagues, maybe I don't know the word, dominate the new tools for proceed this. So I think until the at the end of this workshop, we could have a good plan, a good list that we agreed the how much podcast we could produce. And then just information here about the some past events of this year, where we presented some information from the project. And then we had a soil carbon research symposium that held in Rio de Janeiro, here in Brazil. And we had two biochar 4 in Colombia, and aerosoil last month in Spain. If maybe Tamás could speak some information about this. And here, just to close my presentation, here we have some social media address. We are in Instagram and X and LinkedIn. and we have a homepage that I am put a new information. I was doing this yesterday, but I need to stopped. But I have a lots of pictures from yesterday to put there. And if you, all of you, put information on Instagram or on one of these, please put our name there. And then we could repost. Okay, thank you very much. And then I would like to call the next colleague. Let me see who will be here. Next is Dr. Abad Shabi from France. If he could, just one more thing while Dr. Abad started about the extension. I think I remember right now. I forgot to put my presentation. We are asking for 1 year extension. We are requesting for our project from Brazilian side, according to EJP office. Each country should proceed this by itself, and then we are asking to this 1 year to be possible to modeling team working with some calm, some time to do this all those things they are planning. Okay, thank you very much. Please, Dr. Abad Shabi.
Yeah, good morning, Melissa. Good to hear you. So I will start with the last point regarding the extension, project extension. So I have had discussion with the INR. So I let Natalie know that I emailed you last night about this possible extension, and in principle it's okay. But I just sent the letter because I missed it in between. So I sent this letter this morning asking her this is what we are, and then explaining her the process that each country, and then also informing her from what we receive from Oleika Ziegler that each partner should send the letter independently from the whole consortium and gets the okay for the extension. So I think that is done, and I hope that we will get the extension, which will be very good for the project, but also for everybody, because it will be pity that we end up the project in May without finalizing the last step, because now we are getting the data. So regarding the involvement, so I am with you involved in the coordination and of the project, even you are doing the most of the job. But we don't have in France a specific task. We are involved in different tasks. And then, for example, Cornelia and myself in the work of Joshua, part of the analysis that has been promised to be done in France, now we transfer the budget to Ghana, that in a way that they can perform the analysis. This is something done. I discussed with Caleb yesterday, and then a little bit with Edward, that things is progressing. I'm also partially, I'm sitting close to Joanna, a little bit involved also in the PhD, because we have done some of the analysis for the enzyme activities, and also it's also something that is in the progress, and I hope that we do a second analysis if she wish to do that. Even if it's a little bit expensive, but it's for the project. It will be very interesting to have to complete this set of the data. And with respect from what we have, the database, the historical database, I send you all of this data. What is missing now is the metadata. I'm preparing this, and I hope that I will find time this week to send you the data. We have a new data that we can provide you also. And in the meantime, you remember that for the site that we are proposing within this project, we have done the spatial variability of carbon stock in 2005, 2014, and now we are assessing with my PhD, with Brazilian PhD student. He's in Santa Maria, assessing the calculation stuff, and then we have a very interesting result. That's what happened. A different management practice with respect to grassland grazing, and the release also can be shared within this framework of project. Last night or yesterday, we discussed also with David Whitehead, and then I think this is a very important part. He will speak on that in detail, is the part of the modelization, because this is the part that we need really to provide, and it's also not dependent only on David, but also on the partner, and then who is involved. And then we would like really to see what are the progress also for the young scientists working with you in this regard, and whether we are going to have a specific modelization or just so we can perform a statistical. But David can explain as work package leader on that in detail. So this is so far from my side, and I think that's I think that I highlight all of this progress. I think from my side, from what I see yesterday and from what we heard, is things progressing. Maybe some of the tasks need to be a little bit push, and some of them are more advanced compared to the other. But as you said, that the most important thing is that the commitment of the project is to fill up the database, and then because the last time it was not completely set, and it's one of the weak points that we need to complete, and we need to make sure that the data at the end of the project is there, available, and then for exploration, and then for modelization, whatever we do with this data. Yeah, this is for what I wanted to say for my part, and then if any question, I am here to answer.
Thank you very much, Abad. Thank you. Yes, I received the data. Yes, will be very, very interesting to receive all the metadata.
Yeah.
And the other information about the PhD student work, and this is very, very interesting. And yes, thank you very much. I don't know if the audience would ask something, or may I could proceed and call the next one. Let me just put here, just change my screen here. Yeah.
Good morning. I think it is me.
Am I right? Please.
Yeah.
Thanks.
So I try to share my screen. Okay. Okay, can you see my slides?
Yes, I can.
So I think during the presentation, I'm going to switch off my camera. Sorry about that. So first of all, I would like to express my appreciation to the organizers for this beautiful meeting. I suppose yesterday was an awesome day representing a very high standard. So in this presentation, I try to briefly overview the previous years that we achieved, our contributions, difficulties, and future plans. Sorry, I just read your email, Melissa, in the morning, so it's a little bit longer than 5 slides, but I tried to keep the 10 minutes. Okay, so in nutshell, our contributions. So we tried to contribute as it was requested. We tried to provide the metadata, and as I hope, we uploaded the previous historical result to the Zenodo. Most probably György provided the required data, and finally the consortium agreement was signed, and as I remember, I forwarded to Melissa the signed page by our rector. We contributed in the work package 1, and we are still with György and Yuri, we are contributing in the modeling group. So this is our experiment site. It's a kind of randomized block design with 4 replicates, and there are the size of the plot is a little bit bigger than 200 square meter, and we have 6 different soil tillage practices like disking, shallow tine cultivation, no-till, deep tine cultivation, loosening, and conventional plowing. This is the representing soil of the experiment site. This is an endocalcic chernozem, which is arid, epidensic, humic, loamic, pahic, according to the WRB classification system. We had the harvest last year, and we started the soil sampling on 10th of July, very early in the morning. Probably those who attended to Hungary during the summer could remember that we have very hot summer days, so we started at 4 or 5 in the morning, and we had to finish it before 11 as the temperature reached the 40 centigrades. This is the landscape and the sampling itself. We have, unfortunately, due to the heat, 2 different sampling days, and between the 2 days we had a huge summer storm with a relatively high amount of precipitation. What we carried out in the field, we sampled all the parcels from these depths. We always follow the EJP arouNd protocol compiled by the work package 1. These are the measurements that we performed. Again, we were following the EJP protocol. Let me share some preliminary results from our site. We performed the site and soil characterization. I won't go into big details, but this is the first page of the characterization, and this is the last page of the characterization. And still we had some laboratory results. Now I'm just presenting a kind of shortened version. Here you can see the soil organic carbon stock from the different depths in 3 different cultivations, like shallow cultivation, no-till, and plowing. And for sure we have the nitrogen stocks as well for all the treatments. I'm just presenting here some short ones, and we still calculated the soil organic carbon stock in the POM fraction and the nitrogen stock in the POM fraction, and the soil organic carbon in the MAOM fraction, and for sure the nitrogen stock in the MAOM fraction as well. What we are doing right now, so we are investigating the micro and macro aggregate stability measurements. We are applying wet sieving. We are using the Slake app, which was done by the National Soil Health Institute. It's a kind of very adventurous playground, and we are using laser diffractometry, and also we had the spectral measurements for each sample. This is again just an example providing huge amount of information, and we need a lot of time to digest and to interpret what we see on the curves. So yesterday Professor Vincent just presented the meta publications. This is a photo from the Eurosoil conference from the consortia from the consortium, Tina and her team was there, presented a poster as well. Some future plans. Next Monday, we are going to do our second year soil sampling. We are just sampling the upper 30 centimeter for all parameters. The others will be also sampled, but only for basic chemical and physical parameters. But we have the chance to apply the 16 sRNA sequencing. So that is my dream, that we want to perform this measurement, and for sure we have to, or we must submit an article. We are facing some difficulties as the whole department is forced to move to another building in the city, which is situated opposite side of the city. So everything must be moved there during December or January. Some words about the extensions. Honestly, I didn't receive official information from the national funder. It's a kind of gossip thing. So, but the most important thing that all the members have to declare that we want to go for the extension. They couldn't inform me whether we need a modified consortium agreement or we need a modified task list based on the extension. But in the morning, we received from Melissa the modified Gantt chart as well. And that's it from our side. Thank you for your attention.
Thank you, Tamas. Thank you very much for your brilliant presentation. I think it's very clarified for us. You have a very happy team. I like to see your pictures. The people are working happy.
Because I'm happy too.
Great. Thank you. About the extension, the answer that I received from the EJP office is that this is just with us. I think they are very, how they say, like a problem. They don't see problem. They are not seeing problem in extension. And we need just to send information for them, but it's more important to each of us sending a letter requesting this extension for our funders. So and I provided together with the letter that Abad kindly helped me to write, and you can change and proceed the modification as you wish. This is a kind of a template, and then you need just to put the funder agency name and put together that information about the Gantt chart. But before that, it's very important if all of us agree with the new data that I insert there. I try to put to use all the period of 12 months, just changing, proceed this modification on the time that we need to use to, or I expected to use to provide a paper, a publication, that the time for modeling team, because as I heard from a colleague from Brazil, the more hard work is in harmonized data. So this is very hard because each Excel file, for example, is coming, it's in a different way. So they need to harmonize this and then choose how many sites use, because probably they will use that sites that we have some more variables measured, or not just measured but informed. So they need, for example, precipitation, a lots of information I can't remember right now, but they need information. And if we already have a data set that we have GAD emissions, and that we have different kind of soils and microbiology, they will ask to us to provide, if we can provide some more information. So thinking in this, they will work. I think that's it about the extension. Is that so? We need to just to agree. Dr. Edward, please. Your hands raised. The floor is yours. Your microphone is muted. I think you need to open your microphone. Okay.
Yeah, thank you very much. And good morning to all of you. I would like to get clarity on the extension. Does it mean that each I hope the echo is still there. Hello.
Oh, yeah.
Apologies.
No
I
problem.
want to get clarity on the extension. I want to get clarity on the procedure for the extension. From the narration that you've given to us, it appears each institution is to apply for the extension separately, providing their justification, and we have different funders. So if Ghana, for instance, would like to get extension for our program, would it mean that we would have to go through EJP to the New Zealand, the primary industries, or how should it be?
I am understanding that Ghana is need to proceed or request extension for your funder, New Zealand, GRE, yeah? Yes. And then I think I'm understanding that the Ghana team is almost done, or the tasks. I think, in fact, the team itself doesn't need extension, if I understand in a good way. But if it won't be a problem for you, maybe in this request you need just to confirm that the modeling team from the entire project need this time to proceed or to finish the modeling procedures or the harmonization and data analysis by modeling. So I think using this kind of justification maybe doesn't could be a problem for organic team. Am I right?
Melissa?
Yep.
Yeah, it's Abad speaking.
Yeah,
I know that if they even that they finish all of these tasks and they have done what was required for the project, I think that they need, we cannot say they don't need extension, because if the project want to be extended, that all partner or the funding partner must agree on that. Your argument about the modelization, that they said, okay, we finalize the work and our duty within this project, but of course we need to be part of the modeling exercise, and this was the reason why we need this extension and be part of this extension, even we are not going to have an extra money for that. If one partner skip out and said we don't need the extension, that means the whole project or the whole attempt to extend the project may fall down. So we need all of partner agree on that. Even that they finish their job, because at the end we are as one consortium or as a project with several consortium, so we need to have this okay from everybody because they are part of the project. So I think, Vincent, even if you finish, you would just ask for extension without any cost, and then explaining that your data or the data that you are generating is part of the modeling exercise, and you are involved on that. That's it. It's very important.
Yes, I agree with you. And then I think this
yeah, just the procedure is just send this letter for this what Melissa sent to the funding agency, and then ask for extension without any cost.
Yes, I think this is this could not
a template. Is only
yes, it's by email. I sent by email.
Yeah, and I can just, sorry.
Hi, David.
Hello, Melissa, Edward. I could just explain that if your funding is from the New Zealand GRA, then they will send you a template, and they will ask you to fill in a template, which simply you state your reasons, and then they will reply to you.
Yep. It's easy process. Yeah. You don't need the Zoom because we are already.
Now I can see the audience. Hi. Nice to see you. Thank you. I can see Joanna, Abad, David, Vincent, and some other I didn't know yet.
There's another one. So I said, Melissa, we are here. You can testify that we are here.
Yes, I know. I can't see right now. Oh, closed my screen. I can't see you anymore. Okay, thank you. Okay, I can see right now. Thanks. Now I think if I can move in forward, I would like to.
Hello, Melissa.
Hi. Yes,
Alan will want to speak.
Alan. Yeah.
Hi, Melissa.
Hello.
We are missing you here. We were hoping that you were going to be here.
Oh, I would like to be with you together.
Yeah, I just wanted to also clarify on the issue of the extension. I think Ghana, South Africa, and Namibia, we are all funded by the New Zealand funders, the Ministry of Primary Industries. I've already approached Luke, who is the contact person from MPI, and like David was saying, he sent me a form where it's a simple form where you just need to fill in the details. It's more like an amendment to the agreement that you have as an institution with the ministry or the funder. So it's not a complicated process. You don't need a letter from EJP, from our project, but rather just to tell them that we would like to extend via email, and you send you the form.
Okay, okay, great. But this is do not bring some concerns to you, right? Or for the I'm thinking in a project structure, or about the, how do I say, when we are funding by a funder agency, if you a request for extension, this is not bad for you. It is okay without funding, additional funding. Yes, I don't know if I'm. It was easy to understand me myself right now. I'm sorry. But it is, if it's okay, I would like to say thank you, because as Abad told us, it's very important. All of us asked to this request this extension because the modeling team needs to work with our data. So I think in this way, it's a very good explanation to this request. Okay, I will ask for Alan to provide an update for the project. I know we are kind of best of the time, but I think some colleagues doesn't come, so I think we have time. So Alan, could you give us some update about the project? Now I can't see the audience, I just see myself. So I'm waiting.
Okay. Can you hear me, Melissa? Can you see the slide?
Yes, not yet.
Not yet.
No.
Okay.
I'm just seeing two of me.
Oh.
Yes, I can see.
Now you can see the slides. Yes, I can.
Yes. Oh, great slide.
Can you see me?
They're beautiful. Yeah, not you, but the slides, yes.
Okay, no, that's all right. Good morning, everybody. My name is Alan, and I'm going to be presenting the project progress that we have made for Namibia under the same arouNd project. And in this project, I am not working alone. I'm working with Martha Ikechuku. I'm also working with Nguza Siambango and also Mariek from Namibia Nature Foundation, which is the institution that we have partnered with. But but unfortunately they could not be here due to the costs of traveling. So it's only me who's representing Namibia here. And just a little bit about the project. One of the major challenges that we had was that we didn't have long-term controlled agriculture experiments for conservation agriculture in Namibia. Conservation agriculture was introduced in Namibia around 2005. However, this was mainly being done by government institutions and non-governmental organizations. So what we did is we identified 2 regions that are climate vulnerable or vulnerable to climate change in Namibia. And this is Zambezi region and Kavango East regions. And then in these regions, because we didn't have controlled experiments, what we did was we identified farmers that had been practicing conservation agriculture under the supervision of non-governmental organization, GIZ, which is a German organization, and Namibian Nature Foundation, which is also another non-governmental organization. And these farmers had been practicing conservation agriculture under the supervision of these organizations for more than 5 years. So we used their plots as experimental plots. And so what we actually did is these farmers have 1 hectare that is practicing conventional tillage and 1 hectare that is practicing what they call conservation agriculture. And then we have another hectare of natural land that is adjacent to their plots that has not been tilled or that has not been undergoing any agriculture activities. And just a bit of what we have done in the project. So we have assessed conservation agriculture practices under smallholder farmer managed systems. So what we did was to then look at a slightly different issue. What we are trying to look at is when the farmer is the one who's managing the field, what are the benefits that they are accruing when they say they are practicing conservation agriculture? Because most of the results for conservation agriculture in Namibia and in other Southern African countries are actually showing results from controlled experiments. But when farmers take up this technology, they are not accruing the same benefits. So we wanted to see what is really happening. And we did this in plots where the farmers are growing maize, which is intercropped with cowpeas, and also pearl millet, which is also intercropped with cowpeas. And we have focused on soil quality. We have looked at also focusing on microbial diversity and crop yields. And the objective was to evaluate the medium-term effects of conservation agriculture on soil chemical properties, focusing on electrical conductivity, soil organic carbon, also an extractable phosphorus, ammonium, nitrate, nitrite, and extractable cations, which is sodium, magnesium, potassium, and calcium. And also focus on biological properties, the enzymes, and the microbial biomass, carbon and nitrogen. And in this project, we have 2 master students. We had 1 PhD student that left the project, and so we actually requested from the funder to change again our agreement so that we could be able to get in 2 master students. So we got 2 master students, 1 lady and 1 gentleman, and one of the gentlemen is looking at the changes in soil physical properties, and the other one is looking at the cell chemical and biological properties. These are students who are registered at the University of Namibia undertaking their Masters of Science in Agriculture, and they are currently working in the lab. We have collected our soil samples for the past 2 seasons, and they have been working on generating the results. So just for knowledge advancement, this is the first on-farm scientific evaluation of conservation agriculture in Namibia. So what we are proud of ourselves is that this is the first scientific evaluation of conservation agriculture under farmer-managed systems, and we are looking at enhancing the understanding of how conservation agriculture works under semi-arid conditions and under under farmer-managed systems, and which CA principles matter the most for soil fertility and yield resilience and strengthens Namibia's research base on climate-smart agriculture. We have started generating a bit of some results, Unfortunately, because of the limitations of the number of slides, I cannot share the results, but my two master students, they have generated results, and so far we have one manuscript that we have already fully developed and we have submitted already to a journal, Discover Agriculture, and we are waiting for the review. It's currently under review. And the second paper that we are working on, the master student, the lady, is working on chemical properties. We should be able to submit it by the end of this month, which is within this week. We are going to be able to submit it again to a journal for publication. And we are still hoping to get 2 more manuscripts coming out. So each student is expected to publish at least 2 manuscripts coming out from their experiments. And what we have been observing, I was talking yesterday to Prof about some of the observations that we have been observing in the results, is that under farmer-managed systems, we are not getting much of significant differences in soil chemical properties, mainly focusing on soil organic carbon within the depth of the profile from 0 to 100 centimeters. There is really no difference between when a farmer is saying they are practicing conservation agriculture and a farmer is practicing conventional agriculture, which is most unlikely slightly different from when the conservation agriculture is being practiced under controlled, researcher-managed environments. So we are hoping to communicate these results soon. It might be slightly different from what is really expected, but we are going to communicate the results as they are because they have serious policy implications. We are hoping that the research that we are going to get are going to support Namibia's national climate change policy and also give more impetus to programs that focus on adoption of conservation agriculture and provide evidence for guidance on soil fertility management strategies, and also guide farmers piloting residue retention and reduced tillage for sustainable yields. In terms of collaboration, we have been working with various organizations. We have also visited South Africa as a project at the University of Fort Hare in South Africa. We visited them just to also get to see their experiments and what they are doing there. And also we are also working with the Agricultural Research Council. I will also be visiting on my way back to Namibia next week, the Agricultural Research Council, just to also see the equipment that they have and whether we can be able to collaborate on some of the things that we can be able to do. So basically, this is what we have done so far in Namibia. Our students are hardworking, and we are hoping that they will be able to graduate next year in September, October. They should have been finished with their master's degrees, and hopefully I will also be able to contact some of you for examination of their thesis, if that is not conflict of interest. And lastly, just to also indicate that the students, we are also going to be attending one conference. We are attending a conference in South Africa from the 19th to the 24th of January 2026 in South Africa, the combined congress where the students have papers that have already been accepted for presentation at the conference. And this is one of the outputs that the funder also, we have as part of the deliverables for our project, and the funder is looking forward to us delivering these outputs. So basically, this is what we have done in Namibia. Thank you so much for your attention.
Thank you very much, Alan.
Yes,
sorry.
Yes, I just wanted to also use this opportunity to ask, because one of the deliverables that we have in our project is a review paper that is going to come from the consortium. And I don't know how we are going to work on that, because unfortunately, if we are not able to meet this deliverable, we are not able to get the final tranche of our funding from the funder. And this is a very big chunk of money that we need to get. I think it's about 9,000 euro that we need to get. So I also want us to discuss this so that we can see how we can go ahead on having a review paper that is coming from the consortium, including everyone. Thank you.
Hello.
Thank you, Alan.
Hello, Alan.
Yes, Caleb.
You had a review paper where the whole consortium had to come together. I think you are talking about the technical publication, right?
Yes.
Can you probably, I'm thinking that, is it possible you can negotiate? Because what we did in Ghana was we did our own review and replaced it with the technical publication so that we can move forward. Because if you are going to wait for the consortium to come up with the technical publication,
it's going to be very difficult.
It will be very difficult.
Okay, no, yeah, that's true. I think that would be much easier because trying to get all of us to have a publication, it's not going to be easy. Okay.
Yes, and about technical publication, I think we already have, or is under review, that protocols. I think we have at least one name of from each institution, if I'm not wrong. So Vincent, it's ahead of this part of the deliverable, but I think maybe it could work for Namibia too. But we can discuss this after, of course.
Hello,
but I agree with the Caleb. Yep.
Hello, Meli. We've already, we are in the same shoe as them. We've already submitted a protocol as a technical document, but he is now looking for a review paper. But we were able to do it with the Ghanaian team.
Okay, and the fund accepted that?
Yes, it was accepted by
then that's perfect. That's perfect.
Okay, thank you. Just speaking about the papers, I would like to remind you to put in acknowledgement for the project, the whole project, the arouNd, and the funder. It's very important to mention arouNd in the acknowledgement. Okay, doesn't please remind this.
Yes.
So don't forget to put this in all the papers from the project.
Yeah,
it's very, very important too.
For us also, before you submit the paper, even to the journal, you have to deposit it into a certain platform where the funder has to approve it. So they verify that you have also added this clause that is part of the agreement. There is a very clear clause that you have to include for the funding.
Okay, thank you. Thank you for your update, Alan.
Thank you.
And thank you very much. Now I would like to call Tina. Hi, Tina.
Hello, everybody.
Good morning. I would like to, could you present your update from Estonia?
Yes.
Thank you.
Can you see my presentation?
Yes, I can see.
Yes. So, yes, my name is Tiina Talve. I'm from Center of Estonian Rural Research and Knowledge. And I'm talking about my results during this project, where we studied one long-term field trial and tried to find relationships between soil physical, chemical, and biological properties. Basically, Estonia is located North Europe.
Hi, Tina.
Yes.
Hello, Tina. Can you please put the slide on a full screen? Presentation mode.
Oh, sorry. I have... two screens, and for me, I can see it is full screen.
Okay, then you can continue.
Sorry, I don't know how to switch my view.
No.
I think you can continue.
You can put just a zoom and we are seeing the first slide yet. I don't know if you change your slides, but we are seeing just the first one.
Only first one, okay.
Yes. Now, second one we are seeing. And if you just put just a little more zoom, I think it's you are in 91. You could put 100. Yes.
Is it okay?
I think 100. Yes, I think it's okay. Yes, great.
Can you see now?
Yes, great. Can
Sorry.
No problem.
Okay.
Thank you.
So basically, yeah, Estonia is located quite far away from you, North Europe. We are a small country, and our field trial is quite old one. It started more than 60 years ago, and there has been some redesign of this field trial because of different historical situations in Estonia. But it's located at Kuusiku, North Estonia, and soil is classified as calcis soil with light sandy loam. And in this field trial, basically we are studying two main agrotechnological factors. First is fertilizer with the 3 treatments. One is only mineral fertilizer, moderate dose. Second treatment is a double dose of this mineral fertilizer. And third one is a mineral fertilizer with farmyard manure. Second important factor is crop rotation. The field is divided and one part is only spring barley. It is like cereal monoculture. Second part of this field trial is crop rotation, where we grow 4 field crop rotation. And during this project, we can study a third factor as well, and this is sampling depth. In more detail, we took soil samples in 3 different depths and measured physical properties as soil bulk density, then chemical properties and biological properties. There is some pictures from our field trial and field work. The big picture is took by the drone, and you can see there is spots in the field. So we are taking soil bulk density samples. Shortly, some results. As we see, I show here bulk density, and we can see as expected that yes, soil depth has important impact to bulk density. So bulk density is decreasing by depth, but and there is some differences between crop rotation and fertilization as well. Another example, very important property is organic carbon, and we can see that fertilization has important factor to organic carbon. And of course, depth has organic carbon decrease dramatically by depth. But there is only one table for you. I think it's good to show you that, yes, of course, soil depth is the most important factor, but for us, it's good to compare crop rotation and fertilization. And I can say these are premier results, but fertilization has more impact those measured parameters. But for example, bulk density, it's good to know that crop rotation has more impact to bulk density compared to fertilization. But anyway, I know we have lots of data. We had collected all the data, and right now we are doing lots of statistical analysis. We are writing manuscripts. I hope we can submit the first manuscript this year. But yeah, we are working with this project quite heavily right now, busy writing period. But anyway, thank you right now. That's all for me.
Thank you, Tina. Thank you very much. I'm thinking I cannot open my video because of my host disable my video, but thank you very much for your presentation. And very, very interesting results. It's I took some notes, and it's really interesting as a synthetic fertilizer has so huge impact over carbon stocks on different kind of soils, even temperate soils, is tropical soils. The crop fertilization is still having impact in this. Let me open my video. Thank you very much. Thank you. I like to show my face even at this time of the morning. Thank you. Well, about the extension, it's okay for you, for your
Yes.
team to
Yes.
request.
I already sent this letter, what you send us, and I got extension. So in Estonia, we are fine.
Okay. In Brazil too. So it's great. 1 year, I think it's enough. Oh, sorry, I have 2 dogs here. I'm at home. So I would like to ask. Thank you very much, Tina, for your presentation. I would like to ask for Adornis if I think he is in the audience, so maybe he could present for us.
Morning, colleagues. Hi, Melissa.
Hello, good morning.
Morning. My name is Adonis Niza from South Africa. I present on behalf of the team. There's 4 main members within the team. That is myself, Adonis, and there's Prof. Manyewere, who is also sitting in the audience. There's 2 more colleagues that could make it, Dr. Shira Rubnarian and Dr. Pia Rama, as well as 4 students that we are working with. So I'll provide a few highlights of what we've done so far in South Africa. So we have a trial that was established in 2012 at the Pandolwazi Agricultural High School in the Eastern Cape in South Africa. So it's been going on since 2012. So it's, sorry, I think I put, all right. So it's been going on since 2012. So it's now in year 13. And it was designed to test the effects of tillage, that is no-till versus CT, residual retention versus removal, as well as various rotations such as maize legume or maize cereal. And we were measuring, we're sampling across various soil sampling depths. And for some of the analysis, we used the consortium protocol for sampling and analysis. And my colleague, Prof. Alan, was also involved in WP1 in the formulation of the protocol. And so let me now move on to the key outcomes. We have also managed to deposit 2 data sets into the repository, which contains microbial biomass carbon, enzyme activity, CO2 fluxes, POM, and SOC. We're also trying to sort out some of the data. There was a lot of data since 2012. So I think we still need to clean the data and put it again into the repository. But we have quite a lot of data since 2012 that we still need to sort out and clean and then deposit into the repository. So one of the main activities that we undertake is students training. And so far, we have trained a number of students, 2 MSc students that have completed their studies last year and this year. And then we have 2 students that were still doing PhD that are still studying with us. And we've also brought in 1 postdoc fellow to assist with our research, also to expand the research work, as well as to assist some of our ongoing students. So these 2 MSc students, I think last year, 2020-25, they had attended a conference where they presented poster presentations during that conference, and they've also managed to submit 2 manuscripts, one of which has been published. 2 are still under review, so they've been quite productive. And the PhD students are still in the middle of their studies, but we also hope that they will also draft and submit a couple of manuscripts. And then the 2 PhD students that we have, there's Miss Sanele Poswa and Miss Sinesile Mkengwa. They're still in the early stages of their studies. So Sanele is looking at changes in soil function microbiome diversity after a decade of cooperation and residue retention under CA practices within the Eastern Cape province of South Africa. And to help her with the studies, we sent her for training, for 2 training sessions last year in December and this year on bioinformatics that was in Pretoria, as well as another one which was done in earlier this year, so that they can at least get to understand some of the concepts that will be used during the studies. And the 2 pictures there, they show activities during the training session. It was a formal workshop, as well as the other picture shows some of the sampling activities that they've been undertaking over the past few seasons. And so our second student, who's also doing PhD, she's looking at the role of conservation agriculture in regulating CO2 fluxes in semi-arid regions in the Eastern Cape province. So she's been taking carbon measurements since February this year. So we use a LICO shown in those pictures. And she should also present some of her work next week during a research week of excellence session that will be held at the University of Fort here in the next coming weeks, just to showcase what she's been doing to the university. We've also been working on a number of manuscripts over the past few years. So one of the MSc students published 1 manuscript in soil and tillage research. So basically the work was showcasing the impacts of CA on physical properties. And some of the key findings were that bulk density and porosity were improved by no-till with diversified crop rotation as well. And water retention was also increased by 24% compared to conventional tillage. So these are some of the key findings that we observed during the first 10 years of the study. And second takeaway message was that after a decade of no-till, there was an increase in phosphorus, especially in the top layer, as well as increase of potassium, calcium, magnesium in the subsoil. So this manuscript is still under review. We are hoping that it will be published in the next few months, hopefully. I think this one is in Helion. So far, we received the first set of comments. I think the MSc student is still trying to address the comments and submit for the second stage of review. Then the last manuscript that is under review was looking at the effects of CA on, sorry for that, okay? Effects of CA on mostly organic carbon. So again, we observed increases in organic carbon of up to 14% within no-till systems compared to conventional tillage. There's also higher, particularly POM, as well as improved fulvic and humic acids within CA systems. Again, this manuscript is under review. Hopefully, we'll get a response soon enough. So this is what we've done so far in South Africa. So our students, the PhD students, they're still ongoing. MSc students are finished, and we hopefully because normally the PhD students have been sampling seasonally. So far, they've already sampled for the current season, and they will be busy in the lab over the next few weeks. And I think that's all we have from South Africa. And we also want to thank our funders for the assistance. Thank you so much.
Thank you, Adonis. Oh, great. Thank you very much. I'm very happy to see, to know about that we have so many papers published and so many under review. So I think I will provide you maybe a Google form for sharing this information or to send to me, because this is very, very important to have a kind of register of this, because as we are so many, it's very easy to lose this information. So since our project started 2023, and we have 2 years, so if we doesn't organize this, it's very easy to lost information. So I will provide this form for you. Thank you, Adorns. Let me see the next one according my list here. It's Vincent.
Okay.
Hi, Vincent.
Hello, Melissa. How are you?
Hello, I'm fine. Very
Great.
fine. And you?
Nice
and
seeing
you
you
all?
online. I thought I'd be seeing you in Ghana this time around.
Okay. I would like to see to be with you this time presentially, not virtually, but
Okay, thank you very much. All right, so I want to give a brief update of our work in Ghana. I tried doing this yesterday, so most of you are very familiar with my slides. But Caleb hit me that I have to still do this again today. So I was trying to give him a little bit of no, but okay, so in Ghana, we have about 6 different sites across the country. And as you're already aware, these are the African Dark Earth site in Ghana. So please, can you show my slides? Okay, all right. Okay, so each of these African dark earth sites were selected in 6 locations. And at each of these locations, we had a look at other land use types. And then we have field in Mole National Park. We have field also at Asantequah, which we'll be visiting on Friday or Thursday, I think so. So in Ghana, we selected about 1,000 soil samples. So in Ghana, we took 1,000 soil samples across these areas, which are very hectic. Handling about 1,000 soil samples it's a lot of work. And I must also acknowledge our pedologist, Thomas, for holding the fund for the students. Let's give him a clap for always on field trip with our students. So for the Ghana side, we have almost completed all the analysis, and this is very good news for us. And then we also have a second work in Norway, because Norway didn't receive funding from the funders. So we have to take that up with Germany. So Joshua did a number of samplings in Norway in the snow. And then he took about 160 soil samples in these areas. And I must be happy to say this work is about 95% complete. And then he still wants to do some few analysis on iron and aluminum oxide. So for the 2 PhDs, we are happy to say they are the thesis writing phase. Most of them have finished their lab work. They are just writing. And then the 2 MPhil students, they are 3 actually. Sorry for the mistake. One of them submitted a draft thesis for review, which is under consideration. And during the period, we received an MPhil student from Brazil, from Embrapa, together with Tevino. She did a very important work trying to look at the humin and the humic substances of this African dark earth. And then this will be run to get the isotopic signatures, and very important for this part of the work. So this is more or less chemical fractionation, unlike the physical fractionation that we are doing. So we published a paper recently in soil user management, and Joshua has drafted a paper that will be published in Helion. It's under review. And of course, we have a number of manuscripts also we are working on. So you can see some of the profile pits here in the African dark earth with very thick horizon, and these are purely anthropogenic. And then as we refine these practices, we want to encourage most of the farmers to do a number of work which will also increase the persistent carbon pool. So I showed this yesterday. You can see some of the African dark earth site here. And then you can see a maize crop going very well on this African dark earth with little fertilizer application. You can see Emmanuel Bedu in the photo there taking a field sampling. So we did also outreach programs at one of the communities in northern Ghana where we trained farmers to prepare compost, and there are about 120 farmers. And also, we also established a 10-acre plantation of tree crops. So what we did was we procured the seedlings from Forestry Research Institute of Ghana, and these were given to the farmers to plant. And I'm happy to say most of their trees have really, really established, and they are quite very happy with this. Now, I want to be a little bit brief on presentational results, because the students will do this more. Joshua will come just 2 people after me, and then he did a number of vulnerability studies in the laboratory where he subjected samples to decomposition after a 98-day period. And then you can see that on day 1, there are differences in carbon dioxide evolution from these soils. But surprisingly, after day 1, if you move to day 3, day 7, And to the rest of the days, there were no differences that you can see. And then if you look at this production system, it is purely fodder production system. It is a rotation. So you can see that each of the rotation is 4 years. But then after 3 years of rotation, you will see the soil from those plots having very much emission of carbon dioxide, and it is quite difficult to interpret what could be the cause of this. Because where you have a lot of grassland, you may want to see recalcitrant carbon, but we see more carbon being evolved after 3 years of further production. So I want to conclude that overall the project in Ghana is very much on course. We have made a lot of progress with student work, and also with meeting a lot of stakeholders in terms of conference presentations across the globe. And also the students are at the writing phase of their work, and we also putting a pressure on them to publish and submit their thesis with time. Initially, we were of the view that we may not need the extension, but as we just all agreed, we definitely have to also go for the extension. Then we go along with the consortium. And then we have also uploaded our historical data on time. We were one of the first institutions to upload our historical data. And I'm also happy, Melissa is also very happy about this. And then for our new data, we are almost complete. And then very soon we may be uploading also on the Zenodo platform. So thank you very much.
Thank you very much, Vincent. Yes, you were the first to put data there. Thank you for this. And thank you for your presentation. Very, very good. About the farmers, it's a lot of farmers. I'm very impressed about this. So many people. And it's just a question. I saw that was in one city, but those farmers, they were in the same community. They live together around the same place. Vincent?
Yeah, it's a farming community. So before we were there, we announced our coming. So the chief had to organize the farmers, and then we trained them at their school, their primary school. So they came together, and we offered the training for them.
No, so beautiful work. Congratulations.
Great, thank you.
Thank you very much. Next one, David.
Good morning, Melissa. Good morning, everybody.
Good morning.
Great pleasure it is to be here in this room in Ghana. And I'm sorry, Melissa, you're missing out, but it is a great time we're having here. I would like to take this opportunity to thank the organizers for making this happen. And it's great that we are able to come together. I'd like to update you on what we have done in New Zealand specifically. And I apologize perhaps if it might be a little bit sort of data dense, but I hope very much that if I run through it quickly, it will be of value to see what we've been doing. I just want to remind ourselves, and I've been quite heartened at this meeting here, there's not been a lot of talk about building carbon stocks to mitigate climate change. I mean, when I was part of the 4 per 1,000 group a few years back now, it was very much that we could, of course, solve climate change by increasing soil carbon. Well, it can contribute, but it is never going to solve the climate crisis. And our focus, and everything talked about today, is very much on building sustainability of our agricultural systems, not solving the climate crisis. We must be aware of that. So, and also, I think the point here of these quotations, I leave you to read, but these quotations are that it has often been assumed that increasing soil carbon stocks will increase productivity. That's not always the case. We have to be careful to make these general assumptions. But also the major point here is that retaining carbon is important, but carbon has got to break down in order to release the nutrients and help the microbial processes. We can't just lock off the carbon. We've got to make it available. So that's just reminding us about those terms. Just a quick note about New Zealand. We're a small country. We're a small island, quite different from where we are just here, really. But most of our agricultural land, of which is 53% of our land, is in grasslands. That's the dominant feature. So our focus is on grasslands, really. We've got high soil carbon stocks because we've not had hundreds of years of intensification. But it is incredibly challenging us to hold on to that carbon. To increase the soil carbon is proving really challenging, but we need to maintain it. And that's why we've been we're interested in the vulnerability of maintaining those stocks. And we know there are major impacts of land use on carbon stocks, so that's what we've been investigating. What we chose to do was to show 32 sites across Canterbury, that's shown in the green area, eastern, where I live and are based in New Zealand. And we took these samples of the same soil type, but therefore cropping, dairying, horticulture, sheep and beef on low to rolling lands, and sheep and beef on hill country. Now, I know to many of you, these land uses may seem strange because they're different certainly in many parts in Africa, but they are the major land uses we are concerned about. We've looked at the organic carbon stocks, we've looked at the fractions, we're really interested in carbon loading, and that was discussed yesterday about carbon saturation in relation to how full are our soils. And we've been doing these experiments that Vincent just spoke about in terms of looking at the incubations over long periods of time to look at the vulnerability as well. We've actually been working at different temperatures as well to bring in context of what might happen in a future climate and looking at assessing a vulnerability. So these are some of our data. Now, it's hard for you to see all of these data straight away. So what I've done is just given you the data on the left and the summary on the right. What we show here is carbon stocks. These are, by the way, on an equivalent soil mass basis, not on a depth basis, to account for differences in mass of soil as you go down deeper through the profile. But what it shows is that the carbon stocks in cropping are low. Well, by our standards, they're low. 90 tonnes per hectare in the full profile may not seem low to some people. Horticulture stocks are low as well, but by horticulture in Canterbury, we're talking about low-quality soils with a lot of stones in them because they grow vineyards and they produce wine very profitably. There are higher stocks in terms of the carbon stocks in relation to dairying and sheep and beef on both our flat country and our hill country. So we asked the question, why is there these differences in carbon stocks? And the 2 points we've been looking at are in terms of the fractions. Now, on the left is the plot of the mineral-associated fraction against the total organic fraction. And you can see that there is a very strong linear relationship, but it does tend to level off. This is, we're essentially arguing that TOC can become saturated. MOAC can become saturated. And there's a lot of evidence in the literature. And as we talked about yesterday, there's a lot of controversy around this. But these are what our data are showing. But on the right-hand side, particular organic carbon does not seem to saturate. It can always be increased, providing there are carbon inputs being completely put into the system. And in that relationship, that's a really important that we can still build carbon stocks, even if the mineral stocks are fully saturated. This is carbon loading. We define carbon loading as the mineral-associated component over the mineral surface area, the specific surface area. And this is essentially proportional to how full the carbon stocks are. And again, we've done it by those different land uses. And in summary, there are obviously, carbon loading is high in the upper surfaces because there are more carbon inputs into these systems, certainly into the rooting system down to 30 centimeters. But it's close to saturation. It's close to saturation, almost 1 in our sheep and beef on flat sites. That's really important. We cannot essentially build those mineral stocks any further. But, of course, lower down, there is a lot of capacity to increase those stocks. But the carbon loading is actually lower on the sheep and beef hill country. Remember, I said that they were where the high stocks are, but the carbon loading is lower. And on dairy and horticultural potential, there is potential to increase stocks. The carbon loading is lower. So that's the analysis of mineral associated versus carbon loading. Just to give you a brief insight into the incubation experiments we're doing, on the right axis is the cumulative of carbon emissions from these different soils over a 98-day period at 3 different depths: the top, the middle, and the lower depths. And I'm not going to show you the analysis of mineralization in such a short time, but mineralization over 98 days showed that we've lost 5 to 11% of total carbon stock during that mineralization time. The mineralization rates decreased with temperature, decreased with depth in response to lower carbon stocks. That's not a surprise to us. There's less carbon there, there's less mineralization. But the cumulative mineralization, that's the cumulative mineralization at the end of the period of 98 days, it's very high for the sheep and beef hill country. It's much higher for that land use than it is for cumulative mineralization in other land uses. So there's an interesting process going on in terms of the way we use our land, which is affecting the mineralization of soils. And lastly, I just want to show you in terms of the relationship here between the cumulative mineralization, that is at the end of the 98 days, how much was lost, against particulate organic carbon, because there is a strong relationship, strong enough for us to say that what's driving in mineralization in these soils is not the mineral-associated carbon, which is protected. It's the particulate organic carbon. That seems to be the driver of the losses of carbon in our soils. Again, high mineralization rates for the sheep and beef hill country, but they've also got high particulate carbon and low mineralization rates for the dairy and horticulture sector with low mineral carbon. And I just want to indicate that hot water extractable carbon may be a useful way that we can use to investigate these processes without doing all the fractions and without doing all those incubation rates. It shows there's a very good relation between hot water extractable carbon and the amount of carbon available for mineralization. That might be a way that we could use in future. So just putting all those data together, sheep and beef hill country, they've got high carbon stocks because they've got low mean temperature. They're more elevated, essentially. That's what we're arguing. They've got a higher proportion of particulate organic carbon. They've got high mineralization rates, and therefore they are more vulnerable to temperature increase. They're more vulnerable to land use change. If we were to change that land use, we probably would lose a lot of that particular organic carbon. Then if we go to horticulture, the stocks are low in those soils because they're poor soils. They've got low carbon loading, but they have low mineralization rates. So that carbon is protected, it's not being lost. Whereas in dairy and cropping, lower carbon and particular organic carbon proportion, they've got losses because of disturbance and from irrigation. A lot of New Zealand data show that irrigation actually results in carbon losses rather than carbon gains. And the stocks are very much dependent on carbon inputs. Lower mineralization rates, therefore we would argue that those soils are less vulnerable to temperature increase. So this is the framework we're building up in terms of what's regulating losses and how we can change land use or protect the soil carbon by manipulating land use. So just in conclusion, really, we've been able to quantify soil carbon stocks, and we've been able to look at spatial models. We're now scaling this up to the region rather than looking at our particular sites. In terms of what value is this to policy, well, we'd argue we can identify locations and management practices which are high vulnerability risk. So we can particularly inform land use options in terms of where they might be very careful in terms of changing land use. We can predict the impacts of management practice and the impacts of climate change. We're working towards this in way of extending our data. And we will be recommending specific management practices to retain carbon stocks, reduce vulnerability. And so in the future, well, we're going to extend this analysis spatially to incorporate much more areas. We want to improve our predictions and reduce our costs of measurements for soil properties. But we would very much like to explore the indices of vulnerability in more. And again, working with this group, some of you, as we've heard in Ghana and in Germany, are already doing these measurements of vulnerability. Let us get together and see what we can altogether learn more about the processes affecting vulnerability. Thank you.
Thank you very much, David. Your words are very, I don't know the word for inspiring me. Thank you very much for that. And another thing that we could think is an adaptation about the agriculture, not just mitigation, but thinking in food security. We need to think in being adapted for that. So informing policies, we would think in inventories, I think, these predictions and ways to adaptation. Yeah, agreed with techniques. So thank you very much. Thank you. Next.
Hello,
Joanna.
Melissa. There are some questions
Caleb.
down here. So, Dr.
Oh,
Iwa will
thank
begin
you,
to answer
thank you.
questions.
Yeah.
Please.
Yeah.
Go
Thank
ahead.
you very much, David. I think it's quite exciting to see the figures and the results. I would like to understand your thoughts on building the soil carbon. Now we've seen increases in soil carbon stocks from some of the management practices that you have put in place. But how does it translate into the greenhouse gases emissions? Are we building the soil carbon and reducing the greenhouse gases emission, or you still build the soil carbon and you still have high emissions? I just want to understand from your work how this comes out.
Thank you, Edward. We will have a longer conversation about this. Very happy to talk to you more about this. The New Zealand situation is different, of course, in that most of our emissions come from methane from animals. But we did an analysis and we showed that out of 9 practices which are currently and could be adopted in New Zealand, we realistically looked at what's the potential to reduce carbon, and we looked at the land area where that could happen. And the overall conclusion is we will only ever be able to offset our greenhouse gas emissions by about 3 to 5% over a 20-year period if we were able to increase soil carbon stocks. So that was the point I tried to make at the beginning. It's not going to it's not going to offset our greenhouse gas emissions, but it is going to help sustainability and productivity on a much higher basis. But I would be happy to share that with you later on with a lot more detail, if that's okay.
Thank you, David. I don't know if we have some more questions. Caleb, please.
Hello. I think we are having Johanna.
Ok. Johanna, em português is Joana. Johanna, please.
So, good morning, everyone. My name is Johanna Zimmermann, and I'm a PhD student in arouNd. And yesterday, Caleb asked me to give a short overview of our progress in Norway so far. So, my research is part of a collaboration between the RPTU in Germany and our partner in Norway, the Norwegian Center for Organic Agriculture, NORSOC. So I summarized some of our key deliverables. So for the work package 1, Jeroen gave a presentation at the IUSS in Florence in Italy about the effects of long-term soil organic matter decline on soil nutrient status and organic matter composition and organically managed grass clover lay and permanent pasture in Norway. And he also presented a poster at the last annual Science Days from EJP in Vilnius last year about what causes the ongoing soil organic matter decline in organically managed grass clover lay in Norway. And we recently got accepted a paper, I think 3 weeks ago, so we're still waiting on the publication about the trends in yield and soil quality over the last 30 years of the organically managed grass clover lay in Norway. And we also did some site and soil profile description. We have some harvest data, the soil texture, pH, and so on. I will present to you tomorrow more details about our long-term experiment and some first results that we have. Because we did some greenhouse gas measurements in the field last year and measured some enzyme activity, and a bachelor student of mine did a cold and hot water extractable carbon and nitrogen from the soil in Norway, and I also did some thermal analysis, but I will present to you some of the results tomorrow. Yeah, this is the poster from Jeroen. And for work package 2, we also had 3 posters at different conferences. So I presented some of the, I presented a poster addressing one of the main questions from arouNd, which are the best indicators of soil organic carbon persistence. And I also had a poster this year at the DBG in Tübingen about the impact of anaerobic digested and undigested cattle slurries on soil carbon and nitrogen dynamics under different precipitation scenarios. And I also had a poster at the annual Science Days this year in Denmark about the anaerobic digestion trade-offs and soil organic carbon stabilization and greenhouse gas emissions in a long-term organic field trial in Norway. As I said, I will present some results of this tomorrow. So here are the posters. And the last one I will present tomorrow. We are also planning a column experiment where we are comparing the anaerobic digested slurry and the untreated slurry with 2 different precipitation scenarios, addressing one of the main questions, what are the consequences of extreme weather events on carbon stability, nitrogen cycling, and greenhouse gas emission. And we want to publish a paper about that. And also, like I said, we are currently working on a method paper where I combine the density fractionation of carbon and thermal fractionation to have a better understanding on the carbon persistence. And speaking of collaborations, we are also looking for different soils from all over the world to do that. We have some soil from the US so far, and from Norway and from Germany. But if you would like to collaborate with us, I would be happy about it. And we want to do also an experiment about the vulnerability of organic carbon to decomposition. I think it will be a student experiment, but I think Jeroen can tell you more about that. And we are planning right now a next field campaign where we also take soil samples and gas samplings before and after the slurry application next year in spring. And after that, we would like to publish a paper about the field data. So, thank you for your attention.
Thank you, Johanna. Thank you very much.
Questions?
Yeah, Johanna, were you able to compare some results you have in Norway with that of Germany?
Sorry?
Are you able to compare some data between what you have in Norway and that in Germany?
So we don't have any long-term experiment in Germany in this project. So we are only analyzing data from Norway.
Oh, okay. Well.
Anyone else? Nope. Thank you, Johanna. Thank you very much. I would like to have Joshua here. I don't know if Joshua will present because Vincent speak about some results. Joshua is here.
Yeah, Joshua will be
not here, but here.
Okay, thank you very much for the opportunity. My Norwegian mother, Isabel, was supposed to do this. And since she's not around, I'm stepping in for her. Thank you very much. Okay, so I also present on the update from the long-term experiment in Nibio. And the Nibio is Norwegian Institute of Bioeconomy. So I'm here to present on some few progress we've made so far. And this is going to be the presentation outline. Okay, so this plate shows the field layout. So when you go to the field over there, this is how it looks like. We have 2 reps each, or each farm is replicated twice. So if you pick DAI, for example, the 1 stands for the first rep, and the 12 at the bottom indicate the second rep. So each of the farm has 4 different mini farms in it, or mini plots in it. So that was where the sampling took place. And so each of the system, as I indicated earlier, you see the DAI, which is basically for fodder production. So you could see that it contains about 90% grass and just a 10% for the wheat production. And each of the systems has its features DAI is mostly plow, mostly in the spring. Yes, there is a manual application in collaboration with the mineral fertilizer, and also has a cover crop. Okay, so that is Joshua sampling there. The picture looks like a robot, but it's not a robot because the weather was very cold, so I have to arm myself to deal with the weather. So that is me over there. And one of the difficulties I encountered there was the depth of the soil. When you follow the protocol, it's 0 to 10 centimeters, 10 to 20, 20, 30, 30 to 50, and 50 to 100. But because of the nature of the land over there, I couldn't go deeper. I remember when Isabel asked me, which depth are you going to sample? I was talking, 0 to 100, and she was laughing. She didn't, I'm sure she was saying it in her head.
Kind of freezing.
Yeah. This guy didn't know what he's talking about. But when I go to the field, I realized why she was shaking the head. So this is one of them. You go a little deeper, you meet the rock, so you have to dodge, and then make sure that you scrap everything, then you go deeper. So this is one of the issues seriously encountered on the field. So my report, when you will see it, that is the reason I couldn't go to the 100 centimeters as stated in the protocol. And so after the sampling, the soil samples were oven dry at 40 degrees Celsius because at that time we're in winter, so I couldn't get the normal sun, so we have to resort to oven drying. And then the other one that looked like a pilot over there was when I was milling the plant samples which were collected. And so when the samples were brought back to Ghana, because NIBIO, they didn't have well-established laboratory where we can do it, so I have to ship it back to Ghana, to the soil science laboratory here in KNUST. So the laboratory analysis took place. So this plate shows when I was doing the fractionation. Yes, so that is the picture there. And so after the fractionation, these were the samples that were obtained for the particulate organic matter, which were run to obtain the particulate organic carbon. And so with the physical parameters that we have analyzed so far, include the bulk density, the porosity, and the specific surface area, and then the particle size were also determined. With the chemical properties also, the EC, the pH, as stated here, were all analyzed here. And with the fractionation also, we have the mineral-associated organic carbon and then the nitrogen obtained, and then the particulate organic carbon and nitrogen we also analyzed using the CN analyzer, which also provided us with the ratios as well. And thanks to Isabel, my Norwegian mother, she also provided me with the yield data and also the cover crop biomass yield that were obtained. And so the results from some of the selected, tomorrow, God willing, I'll give update on the most of the results we've obtained. But for the want of time for today, I would like to state some of the results that were obtained. Here we have the particulate organic carbon, which were also high, mostly across the grass land or grass plot. So when we pick the mineral-associated organic carbon, look at DAI, the third-year grass. It gave a highest mineral associated organic carbon, which I'm tempted to believe that, yes, grass also help with the stabilization or with the carbon improvement of soil organic carbon, but the longer it is on the field, the higher the contribution of organic carbon. Because when we look at this data, we have grass which are 1 year old, another 2 years, and a third year. But when you compare these three grasses, the third year grass was given high organic carbon compared to the rest. And also the bulk soil nitrogen were also obtained, and the same trend was observed. Now, with the contribution of the pools to total organic carbon, it also indicated that across all the systems, they all gave contribution more than 60%, which means that most of the carbon were stored in the mineral pool, which is a very good news for us, because we know that if we have most of your carbon in the particulate pool, within some few years, it will all get mineralized, and the crop may suffer at the long run. For immediate effect, it is good, but for the long term, it's no good for us to have more of our carbon in the particulate than the mineral associated. And with the same trend, when it comes to the contribution to total nitrogen, it also follows similar trend, which is far above 50%. So most of them were within 70 and 80% mineral associated organic nitrogen and its contribution to the total nitrogen. And so the soil carbon stocks also indicate, my professor yesterday mentioned this in his slide, that as the surface area increases, we also have the stock also increases, which is more or less like a positive correlation for us. And so with the yield also, we have the grasses also given us. Now, before that, the crops, the weight or the drying or the moisture content at which they were taken differs. So when you pick potato, for example, because it's mostly sold at the fresh state, the moisture content was about 85%. That is why I didn't include it in this graph, because when I added it, it was making the rest look not significant. So I have to reduce it for those ones who also have each way. Now, the outstanding activities that we are here to look at with the emissions. The samples have been sent to Kenya for the analysis. So they've sent the first batch from the first day to the 35th day. So we are expecting the second analysis from them, which will be from the 56th to the 98th days. And also the analysis are yet to be conducted. My boss also mentioned that we are yet to do the aluminum and iron oxide. So we are yet to do those ones too in the lab. And then we'll proceed with the manuscript writing. So by concluding, I want to use this opportunity to thank EJP Carbon arouNd and to my supervisors here in Ghana and those from outside, including Dr. Abad and my Norwegian mother, Isabel, for their contribution towards this. What she taught me was to say "tusi tak," meaning that thank you 1,000 times.
Thank you, Joshua. Caleb, we have some questions from audience.
Joshua, from me, a very good presentation. We'll have to look at your data and do some analysis. But when you were talking about the carbon, I realize where you are coming from, because in our tropical system, we have a lot of fire. So we tend to have a lot of our carbon in the forested area than within the carbon system. And then within the forested grassland, you also have much more stabilized carbon. But in the European system, we tend to have more carbon within the grassland. And then when you look at their buildup, it is even much more higher than what we even have within our forested system here in the tropics. So it is something you will have to sit down and then we build upon in your discussion.
Thank you.
Yes, and about the...
Hello, Melissa. Alan will
Another question?
Alan will want to talk.
Okay, thank you.
Okay, I just wanted to ask Joshua about the sampling to 100 centimeters. Were you digging with a shovel or were you using a soil auger?
I was using an auger.
Oh, okay.
Yes, please. But I dug that to demonstrate something, that there was something that was the reason I couldn't go deeper.
Any other questions?
Yeah, I will do. A question from Audu.
Hello, Joe.
I don't know if...
Hello, Joe.
Yeah.
Yes. Thank you for your nice presentation. From the figures, you had land use systems, and the objective was to see differences between the land use systems. That is fine. But when you presented, means were not separated. You only presented the mean values. And when it's like that, it is difficult to compare things.
Thank you. You see, the graphs I showed on the slides, you will see that I indicated that the error bars is equal to the standard deviation. So we can equally use those things for checking the differences in it. Thank you.
The error bars can be equal to the standard deviations, all right.
What he was just doing right now was to focus on the distributions. He will be doing a proper presentation.
Maybe a trend, but the error bar cannot give you the mean separation. You have to use a tool.
Yeah. Meaning you have to go...
Like use post hoc.
Yeah.
Yes, to separate the means. But error bar is a way of showing, predicting error margins for you. Yeah. Okay.
So I think what he's going to do is tomorrow he's going to do a deeper presentation.
Thank you.
Okay. Melissa, do you have something for us?
I have just a question about how deep he dig, because I didn't catch this information.
Okay, I sampled 50 centimeters. So I couldn't go 50. To the hand. Yes, please.
Oh, yeah. Which type of soil?
It's cambisol.
Cambisol. Oh, yeah. So it's correct.
Relatively young soil. So.
Yeah. Okay. Okay, thank you. Thank you very much. The next
Melissa,
will be, yeah,
Jerome will be
come.
coming, I think, in about 2 hours' time. So, okay, Johanna will give us more information.
Yeah, Jeroen landed, I think, about 20 minutes ago. So he will be here soon, but I think he will not make it until the coffee break. So maybe he will do his presentation tomorrow morning, if that's possible.
Yes, of course. Of course. No problem. I don't think so. Agustin or Claudia is coming, or they are not in Ghana, and I think they are not here virtually. Agustin is still. We are still organizing our. How do I say? They are organizing the information about the agreement. They are worried because they not receive funding. So they are very happy to contribute with data, but they are still not put this in agreement or addition. But I speak with EJP office. There are no problem about that. We are just agree between us. So I'm waiting for him about it, but I will try to ask to him again. So because of that, they are not be here. And Claudia, she sent information. She sent data to Zenodo repository. So we already have data from Canada, and they are working on microbiology methods, and they are discussing information and trying to provide some results. But we already have lots of results from Canada in Zenodo. So I think that's it about Canada. Yeah, I think the next step for the program, it's coffee. I think a lot of you are needing coffee right now. So I think after coffee, maybe I could start it with just pick some information about each presentation. And then I think Caleb and the team will separate the presents in different rooms, small rooms. Yes, Caleb, if you would like to give some information.
Melissa, I think for the breakup rooms, we are looking at an opportunity where we'll have a lot of us here to make it much more effective. So we've also discussed a whole lot of the things we need to do. WP1 is almost 100% done. When it comes to WP2, we are still within the primary stage, so there will be not too much to discuss. WP3 too, most of the activities we'll do in WP3 will be met here. So probably, I'm thinking in the next round, Adonis, is it possible we could execute our podcast plan for the partners here? Is it possible? And then you can participate online whilst all those things are going. We may set up a seat in front, and then we execute that particular plan. What do you think?
Sorry, my microphone was muted. I'm thinking great to discuss podcast plan, and because I think if you were divided in small rooms, I couldn't participate. So I think here I can't. I can't. Yeah. But so great.
But if it is possible to, maybe when we come back, you can help us to maybe give us a brief information about what WP1 needs to do next, WP2, and then WP3 before we do the podcast.
Yes, after coffee.
After coffee.
Okay. Okay. So we have 15 minutes of coffee. Yes. Kayla? I can't see any of you. I'm just seeing me. So, now it's 7 a.m. here. Okay, now it's 10 to 11 for you. And maybe we can come back 11 and 5. Is it good?
So, Melissa, Ghanaian
Yes.
time, UTC, probably we come at 11:10.
11:10.
Yes.
Okay. So for me, 8:10.
Yeah.
Okay. Okay. I will take note here. 8 and 10. Okay. Okay. See you soon. Have a good coffee break. I will take mine too here.
Okay. See
Not
you.
so good as yours, but see you soon. Bye-bye.
Already chosen, I know who I am. I know what you've spoken. I'm already loved more than I could imagine, and that is enough. Oh, I'm already loved. I'm already chosen. You spoken. I'm already loved more than I could even imagine. That is enough. It's enough for me. It's enough for me. That is enough. That is enough. Every word you've spoken over me. That is enough. That is enough. All your promises are yes and amen. That is enough. 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 You are enough. You are enough. You are enough. You are enough. So I am enough. Yeah. I believe it. 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 If he dresses the lilies with beauty and splendor, how much more will he clothe you? How much more will he clothe you? If he watches over every sparrow, how much more does he love you? How much more does he love you? If he dresses the lilies with beauty and splendor, how much more does he love you? How much more will he clothe you? If he watches over every sparrow, don't you worry. Don't you worry. How much more does he love you? according to his power, working in us. How much more does he love? It's more than you ask, think, or imagine. According to his power, working in us. How much more than he loves? It's more than you ask, think, or imagine. According to his power, working in us. How much more does he love? How much more does he love? How much more does he love you? How much more does he love you? How much more does he love you? How much more? How much more? How much more does he love you? How much more does he love you? How much more does he love you? We call you Jireh, 'cause you are enough. Oh yeah, you are, yes you are. Jireh, you are enough. So I will be content in every circumstance. Oh Jireh, you are enough.
One thing I desire, only this house, just to dwell. This will be my posture, laying at your feet, just to dwell. Let's sing aloud, church. Dearest Father, closest friend, most beautiful, most beautiful. Dearest Father, you're our closest friend. You are most beautiful. You are one thing I desire. Only this I seek, just to dwell. This will be my posture. I'll lay right here at your feet. Oh, just to dwell here forever. We know who you are, dearest Father. You're our closest friend. You are most beautiful. Hallelujah. You are dearest Father. I know you to be your presence here, your glory here. Dearest Father, you're our closest friend. You are your glory. Dearest Father, closest friend. You are most beautiful. One more time, let's sing it, church. Dearest Father, you're our closest. You are. I know you to be most beautiful. Dearest Father, you're our closest. You are most beautiful. That's all we have left to offer. Oh, sing it loud. There are no words to describe your goodness, Jesus. Our love will sing to you. Oh, there are no words. There's nothing left. Our love will sing. Our love will sing. We'll sing. Oh, to describe your faithfulness, Jesus. You've been so good. You've been so good. We sing. Oh, there are no words to describe your consistency. So our love will sing. Our love will sing. We sing. Oh, now somebody just lift your worship in the room. Come on. We worship you, Jesus. We worship you, Savior. We worship you, healer. You've been a good friend. You've been a good, good father. We love you. We honor you. We adore you. We adore you. There's nobody like you. You're holy. You're righteous. Majestic. You're perfect in all your ways. So there are no words. There's nothing left. Yes, our love will sing to you. There's nothing left. So I'm so in love with you. You're beautiful, so beautiful. We've seen your goodness in the land of the living. I fix my eyes. You're beautiful, so beautiful. If you've tasted and seen the goodness of the Lord, say, I'm so in love. You're beautiful. There's nobody else like you. You have eyes like fire and hair like wool. I'll fix my eyes on the author and the finisher of my faith. So beautiful. With just one look, everything changes. I'm captivated, and I'll never be the same. With just all it takes is one look, everything. I'm captivated, I'll never be the same. Just one, everything. I'm captivated. I'll never be with just one look. Everything changes. I'm captivated. I'll never be. One more time, say, with just one. Everything. My body was healed with one look. My mind was regulated with one look. With just one. My family was rescued with one look. My baby was cured of cancer with one look. Just one. All it takes is one. All we need is one. All we want is just one look, just one. My family was rescued. My children were saved. Yes, they were. I got all the mercy and grace that I need with just with. My mother was healed from cancer. My grandfather was healed from diabetes. Your family was rescued from sin and shame with just everything changed. I'm captivated. I'll never be. I'm so in love with you. You're I'll fix my eyes. There's no one else like you. You're beautiful. One more time, just the voices. I'm so in love. Oh yeah. So beautiful. Your grace is beautiful. Your mercy beautiful. I fix my oh, so beautiful. With just everything changed. Just one. Everything changing. I'm not leaving the same. I'm not leaving the same. I'm not. Just one look. Everything changing. I'm captivated. I'll never be. That's just one. Father, we acknowledge your presence. We acknowledge not just your presence, but the power of your presence. That we can be here and come in one way. And because of your love and your grace and your mercy and your presence, that we leave changed, that everything that's attached to us changes, that our lives change, our families change, our homes change, our jobs change, that our perspective changes right here in your presence. Our shame is removed, our guilt is removed. Our hearts are mended in your presence. Your presence changes everything. If you know his presence is a life-changing thing, would you lift up words of worship and words of praise for his presence, for his presence, for his presence, for his presence. In his presence there is fullness of joy and pleasures forevermore. In his presence there's liberty. In his presence is his spirit. In his presence there's healing. In his presence there's joy. In his presence there's salvation. We thank you for your presence. I'll never be the same with just one me. If the altar's where you need us, take me there, take me there. If you're looking for an offering, I'm right here, my life is here, and I'll be a living sacrifice for you. You're refined, you're refined. I wanna be consumed. I wanna be tried by fire, purified. You take whatever you desire. Lord, here's my life. I wanna be tried by fire, purified. You take whatever you desire. Lord, here's my life. If your glory wants to come in, let it fall. We want it all. Lord, your fire is consuming. Fill this place, set it ablaze. Now we are living sacrifice for you. You're the fire, you're the refiner. I wanna be consumed. I wanna be tried. Yeah. Purified. You take whatever you desire. Lord, here's my life. I wanna be tried. Purified. You take whatever you desire. Lord, here's my life. I wanna burn for you, only for you. Take my life as a sacrifice. I wanna burn for you, only for you. Clean my, purify my heart. Only for you. Take my hand as a sacrifice. I wanna burn only for you. To clean my hands, purify my heart. I wanna burn only for you. Take my hands as a sacrifice. I wanna burn for you, only for you. Clean my hands, yeah. I wanna burn only for you. Take my life as a sacrifice. Only I wanna be tried by fire, purified. You take whatever you desire. Lord, here's my life. I wanna be tried by fire.
Hello, Melissa.
Hello.
Yeah, we are 10 minutes late.
No problem. The people, are the people coming yet? Or all of you are in the room?
Is your faithfulness to me? And I'll still bless you. Yes, I'll still bless you. Yes, I will. Yes, I will. Yes, I will. Yes, I will. I'll still bless you. I'll still bless you. Oh, yes, I will. Yes, I will. Yes, I will. Yes, made a decision. I'll still bless you. Oh, I'll still bless you. Yes, I will. Yes, yes, I will. Oh, great is your faithfulness. Great is your faithfulness. Great is your faithfulness. Great is your faithfulness. From the rising sun, to the setting, same I will praise your name. Oh, great, great is your faithfulness to me. Morning by morning, mercies I see.
Hello, Melissa.
Hello.
Please, you can begin.
Oh, great. I will share my screen. Can you see my screen?
Yes, we can, but the words are very small.
So let me try another way. Oops. Here. By parts. Just a minute, please. Oops. Not so big. I'm home and you are hearing my dogs. I'm sorry. I have two. And sometimes they.
Melissa is about to print, so please.
Thank you. Here, let me share. I think I will try to share right now without presentation mode. Let me see if you can see better. I don't know what I did here with my window. but you just, I'm not seeing you. Let me share what do I, okay. Ah, yeah. Oh, it's terrible. May I try using Excel file? Just one more minute. I'm sorry. Let me close here. Let me open Excel file first. I think it could be better because Excel is Excel. Now, can you see? Let me increase. Okay. Can you see?
Yeah.
Now, can you read? More important.
It's difficult still, but we can manage. It's getting better. Yeah, that's good.
Okay. Yes. Well, I will read. Yesterday, I sent for the team a new an updated Gantt Gantt file. Let me see here. Well, here. In fact, this is not the last one. I'm sorry. Let me try to open the other here. Okay, but here we have two deliverables in in the not the first, but the work package. Let me see, 0. Here, the management and coordination project. And here we have two deliverables. Agreement signed. This is almost. We still we still are missing the Uruguay, INEA. It's just this institution is missing. If you, I don't know if some of you know directly Agustin or Andres. If yes, you could try to access this, but but if not, I'm trying to by email. And the another deliverable is the protocol for data use, flow, and security. This is under review. This is done. This deliverable is almost done, but we will publish it because of that. This is under review. So we will publish by Embrapa. So they are in process. These are in process. So I put this agreement signed for until we are in October. I hope until end of November we have this. Because we are just missing one signature. So in fact, this is very simple, but seems not so very simple. But I'm trying to. And the other, I hope so at the beginning of the next year. This is for the next step. For the work package 1, we have two deliverables. Sorry, We have 2 deliverables. The protocols for soil sampling, this is done too. This was done. And we is in the same way as the first one, another protocol. This will be published. This is under review too. So we are just filling some information because we are publishing under Embrapa series. So we have some, how do I say, normas in English? I don't know if some of you know, but some rules about the form. So we need just to achieve this. So this is the same stage at the beginning of the next year for the organized and harmonized and accessible data sets. This is, we almost have this. Just a minute. Problem solved. Toy was putting inside of my laptop. This is for you. This is the cause of the war here. Okay, I'm sorry. But the data sets, as I told you, we already have 21 data sets in the repositories. The next step is we have a colleague here from Brazil. He have a team. He is working in the Embrapa Agricultura Digital, so digital agriculture. And he has a team of students which is working on pick the information directly from Excel file and through, I don't know if Python or R software, and using some techniques from those expertise. And they will put this information in the information that are needed by the modeling team work. So we are starting this phase. So we are discussing, I think in the next meeting, general meeting, maybe our colleague Luis Barioni could update us about it. But until now, we are exactly in this point. If I'm not wrong, David was in one of the last meetings, and then I think if I'm not forget anything, we are in. So we are in this time. I think here from Brazilian side, we are still missing some data sets. So a colleague of mine who are participating of this project, and she is working with GAD emissions in paddy rice system, and she has lots of data, but she is having some concerns to have time to proceed this upload. As I told you, we have lots of this the same situation, especially from here. From abroad, as I'm not wrong, we are just missing information from Uruguay, and their site is the oldest one. It's from 1963, so it's very old. It's the oldest one in our consortium. So here from this work package, from the deliverables, we are just missing this data set. And from here, we need to proceed the harmonization, still organized, but harmonization, this data set to start to work from the next work package. And let me see here. And for this happens, or publication of synthesizing findings in meta-analysis of regional drivers of soil carbon stocks, and summarizing the findings of management practice and the vulnerability of soil carbon stocks to loss. For those things happen, we need the first step, we need the modeling team. So because of that, is the next step is focusing. I think our energy needs to be in this information, especially for those colleagues from Brazilian side who didn't upload the data there. And if some of you are still missing information, or would you like to add some information to your data set, if you have, for example, data for precipitation or solar radiation, if you have a meteorological station near from your station, from your experiment, is very important for modeling team to have this information. So we could focus in this, put those data there. I think this is very important. And for the third work package, I think we achieved lots of things. We achieve from the below to top, the project logo, web design, and social media appearance we already have. Reporting, we are doing this times and times for EJP. So we have to send partial reports to there. We are sending, and we are just in the first global workshop. I think until the end of the event, we can provide this, the final global workshop, and the money we think after. But so we will think in the one more. Few days, lots of you are doing in your countries, but tomorrow or the day after tomorrow, and the program is planned a few days, a very interesting few days. So done too. And technical publication, we already have 2 under review, because those are technical publication. But here, I think we could include some publications to be consumed by farmers. So we could deliver information that they could use directly. For example, I'm sorry, that about that techniques that were delivered to the farmers by presented by Vincent. So that technique we could produce a 1-2 page technical publication. How to do it? I don't know if they already did it. I don't know yet, but this could be using a very a very simple, a different language, not scientific language, but a popular language. And then we could, in fact, we could deliver this information because it's very important. They really understand and they really take this technique as the technique. It's mine technique. So I helped to develop this. I tried it in my field. So if possible, they could be involved, to some of them, of course, but to be involved in the discussion of this publication before we published. So they could act as a reviewer, something like that. Yeah, just to know if the information is okay, it's understandable. So I think we could provide at least one technical publication per country. This is it's not so much. It's very few, but at least I think one per country could be very, very interesting. But it's not mandatory. We need to be to have at least one. But as David was as David told us, we are just concerned about greenhouse gas emissions, or we are concerned about food security. So I think spreading these techniques, we could achieve both. So we need to have a balance. We need to provide food, but preferable using great techniques, because we already have these. We need to deliver, just to deliver. And if we could add to this technical publication some information about how much money they could save, or they could have by how the word for this profitability. I think they could hear us better. Yeah. So I think this is the main information that we have to have in our minds in the next months. We almost achieve all the deliverables. We just having to request more time, just because the harmonization and of course the final publication, but this will run kind of together. So the other things, I think the team had a great job. So I'm very happy to see this, and I'm very proud of the team. So I think we could discuss this next step, including here this podcast, as Caleb suggested. Maybe we could using the same for the technical publication. So each podcast could be directly linked to this material. So maybe we could do solve this problem or this deliverable together. So would you like to, someone would like to speak? Thank you very much.
So, Melissa, I heard you talking about the training that our Ghana team organized for farmers in northern Ghana. A brief write-up, if I got you right. We submitted a 2-page report to the funder. That was last year with some pictures. Is it something similar that will be required here, or is it something in a different language? Because I heard you talking about that outreach program.
Yes. This technical publication, even the podcasts, we agreed that we could provide it in different languages, for example, in Portuguese, in Spanish, in English, mainly English for be spread, but in our own language to achieve the farmers. Because, for example, here in Brazil, almost none speak in English. They speak in Portuguese. I understand that maybe the same for some other countries. I don't know. Maybe the conference could show the audience. Is it possible just to move the camera for the audience?
Oh, okay. Melissa, they will be working. Okay.
I'm just seeing me.
Yeah, I hope you can see us. I think they have to.
Not yet.
Control room. Tell the control room. Joshua. So, Melissa, what is the next step now?
Next step would be discuss the podcast list. I think here I show you the deliverables that we agreed at the beginning of the project, and we are achieving almost all those. So we already have work to provide to do in the harmonization data, but is not directly from us because in fact I don't know how to do it. But we have a team working with modeling with colleagues from Ghana, from Hungary, Brazil. I'm missing Raya from France. She was participating from the modeling team at the beginning, but we can't. She won't be able, I don't know, to participate for the in these meetings. So maybe Abad could ask to her if she is interested, or if Inhai has another scientist who would like to work on it. Would be very, very good. We have
Melissa?
Germany.
Melissa?
Yes.
You mean in France somebody, Andrea?
Yes, she I don't know. I don't have news about her. And
So I will ask Cornelia because I'm really not aware about that, but I will make sure that this person is. I don't know. I don't know. The first time I heard about this lady. So anyway, I will ask Andrea, Cornelia about this
okay.
this person and let you know.
Okay.
Yeah.
Okay. Thank you very much.
You're welcome.
Because
welcome.
it's
Yeah, you're welcome.
yes, it's very.
Yeah.
It would be very good, very nice to
Yeah,
have
I will let you
a
know.
colleague
Yeah,
from
yeah,
there.
yeah.
Okay. Well, we have from Germany, Hungary, Brazil, Ghana,
New Zealand.
and New Zealand, David. Yes, and if one of you or another country would like to participate directly of this modeling team, would be very welcome to participate. Just tell me or send me an email, and I will invite you to participate of the meetings. And yes, so I think all those things are running. And what we have to provide is some more information if we have directly in the repositories as soon as possible. And provide, now I think we could discuss this podcast lists and subjects and who would like to participate or who could do it. It's not so complicated. As, for example, I'm speaking with you in the morning using my laptop, and this podcast is without screen, are just speaking, and we could provide a script. So in the for shy people, we have just to read. It's I think it's a way, but it's very very it's easy, and we will do it between friends. So it's it's a very funny, in fact.
Melissa,
So, yeah.
if I may come in, I've got a sample that I did with Namibia, so maybe I can share that with the audience for their inputs, and if you like, we can have a few volunteers to continue with the podcast. If that's okay with you.
Yes, I'm okay if the floor was yours, because I think the script is not mine. I just show to you at the beginning of the day. So it's very good if you could discuss directly, and I'm just take notes and giving my opinions if you want. What do you think?
So, Melissa, he already has it printed, what he has to say within 6 to 10 minutes. So what we do right now is to execute the plan. Adonis is already having, so we'll start with the partners available, and then we do it. Luckily for us, we also have the PhD students, so it makes our work easier. And then we have one of our media personalities who did some interviews. Some of all these interviews they did for our national televisions and news media can also be given to the group. She is here. That is Anita. And she is also going to help Adonis to complete this task.
Great. I think you can follow this using all those teams. All this team, it's a very strong team, and then we could, I think, how this work package is coordinated by Ghana.
We are doing it with Adonis and Melissa.
Yes. Yes. So,
I think-
Adonis,
I think
could you?
I think what we can do, I don't know if I want us to do it here or we can find somewhere to do it. Here. Yeah. That's what I thought, but if you want to, because maybe some people I'm more comfortable to.
This we could use this workshop to provide some interviews. That's it. I don't know if I understand well about Caleb told us, and these interviews with these rock stars that are in the room, and the interview could be material for us in the future. And the podcast, we could plan this, but the podcast could be more about the techniques, the results per se, more than subjects. What are you thinking? Because let me see my presentation here. What the initially planned was here, the structure.
So maybe let me run through the questions in the midst, so that at least you are aware of what we are asking. Just a few questions, simple questions about. So the first question will be on. We ask you to give us a brief background of the project, and then you also mentioned the key milestones that you have achieved so far, and then highlight any interesting challenges or discoveries that you have encountered. Then plans for the way forward, and then any knowledge transfer and stakeholder engagement that you have been undertaking. So just a brief update of your activities in your country, more like the same as what we presented during the project presentations. Is that clear, or is that okay with you? The questions, just simple stuff to update people on what you're doing. It's fine?
This is kind of the people presented today.
Yeah.
Yeah, very interesting. From each partner that, for example, from David, from Abad, from the other colleagues who presented today, for example.
Okay, so just to see how it goes, we can start with Alan. I think Alan seems to be ready to. Yeah.
Alan? Alan could start? Yes?
Yeah.
Oh, great.
We need another microphone. Yeah, just come.
Is the microphone working? Yeah, I think.
Hello and welcome to the arouNd podcast. My name is Adonis and I'm happy to have you here with us. For those of you joining us for the first time, the arouNd project is a collaborative project focused on refining soil conservation and regenerative practices to enhance carbon sequestration and reduce greenhouse gas emissions. So our aim is to enhance management of agriculture, both short and long term, effects of soil organic matter persistence and how these insights can inform national inventories and global climate policy. So in this episode, we will highlight the incredible work done by our partners, especially their contributions to Work Package 3. You will hear directly from them about their activities, achievements, challenges, and future plans. So today, we'll start off with Namibia, and with me is Dr. Alan Mpambra. Welcome to the podcast.
Thank you very much for having me.
So Dr. Mpambra, may you please highlight the background of the project, so what you're doing in Namibia for the audience?
Thank you so much, Adonis, for that. So, in Namibia, we had a very unique experimental setup because we don't have a long-term experiment that has been monitored by researchers. So what we ended up having to do was to use farmers that were working together with non-governmental organizations, practicing conservation agriculture for more than 5 years. So we went into these farmers' fields and used their plots as experimental plots. And in these fields, we established 1 hectare that they have been practicing conventional tillage, 1 hectare where they have been practicing conservation agriculture, and 1 hectare which we considered as no-till or as the natural land where they have not been doing any agricultural activities. So this is what we ended up having to use as our experimental plots. And for that, we have been collecting soil samples for the past 2 seasons. So in brief, that's basically what we have been doing.
Thank you so much for that. So what is your focus within the project? What is the main focus of the project?
So we are focusing on 2 aspects. We are looking at changes in chemical properties, focusing on changes in electrical conductivity, pH, organic carbon, and the cations, sodium, magnesium, potassium, and calcium, and also an extractable phosphorus and nitrite. So we are looking at these chemical properties and how they are changing due to the different tillage systems, which is conservation agriculture, conventional tillage, and natural land. And we are also having to focus on changes in microbial and biochemical processes. We are looking at enzymes, changes in enzymes that mainly drive changes in carbon and nitrogen and phosphorus in the soil. So we are also focusing on that. And we are also looking at changes in soil physical properties, which is bulk density, penetration resistance, among others.
Okay, thank you for those highlights. So what milestones have you achieved so far?
So milestones, I would basically say, first of all, we have managed to recruit students. So we have 2 postgraduate students that are working on the project. And we have Elizabeth, one of the master's students, and we have Silas, who's also another master's student. So they are all working on the project. And so far, they have managed to collect data from some of the soil samples. We have managed to analyze them for the chemical properties, and we are currently in the process of writing manuscripts. And we have also undertaken a survey just to understand the relationship or the understanding of farmers on the issues of conservation agriculture, and we have also already worked on the data from that particular manuscript, and it has already been submitted to a journal. So our students have also presented their proposals to the School Higher Degrees Committee. So their proposals have been accepted. So we have made great strides towards the project, and we are hoping as we come towards the close of the project, though there is prospects of extension, we will be able to meet our deliverables for the project.
Okay. It seems like things are moving well for you, but have there been any challenges that you've encountered?
Yes, we did encounter challenges. First of all, we were supposed to have recruited a PhD student for our project, but unfortunately, the PhD student that we had recruited left for the project, so we had to amend our grant agreement with the funder so that we could be able to recruit the master students that we now have on the project. So that was the major hurdle that I would say we faced on our project.
All right, thank you. Then, so what next after this?
So we are looking into, we have finalized the soil samplings, and we now have all the soil samples collected and processed, and the students are hard at work trying to characterize them for the various chemical, and we are done with the chemical aspect. We are moving into the biochemical properties. So we are looking into finalizing the data collection and then publishing this work, submitting it to journals so that we can also publish it, which is a requirement for the students to also graduate. And eventually, the final milestone is to see the students graduating, and we are hoping that they will graduate next year, 2026, by September.
Thank you. So one of the objectives is to share knowledge with stakeholders. It could be scientists or farmers or any regular person that may be interested. How have you been engaging with stakeholders within the project?
So we have engaged stakeholders both at farmer level and at, I would say, researcher level. We have presented some of the work at a local conference that happened in Namibia, and we are also hoping to present this work at a conference that is going to happen in South Africa in 2026 in January. And also what is important is that we have collected our soil samples from the farmers themselves. So we are going to go and give them feedback. We are going to have like a field day where early next year we are supposed to go and present the information that we have observed from the farmers' fields, because they are eagerly waiting for these results, because they would like to see whether what they've been doing has improved their soils, what has happened, and what are some of the recommendations that we can give them. So we are going to engage the farmers. We are going to provide them with the feedback on what we have observed from their fields as part of the research that the students have been taking.
Okay, thanks so much, Alan, for the updates, and that brings us to the end of the podcast. And as we recap, I think we've heard from Alan what they're doing, what they've achieved, the challenges that they've encountered, and how they managed to overcome the challenges. So if you want to get more information on the arouNd project, you can visit our website, follow us on social media, and keep an eye on future episodes for the podcast. There'll be more members participating in this podcast as we go forward. And thank you so much for being around. And until next time,
Thank you so much, Adonis, for having me. Thank you.
Okay, so our next guest is David from New Zealand. Welcome, David. Thank you. So I won't go into the background. I'll just go into it since we've already introduced the purpose of the podcast and the background. So, David, let's start with your background, your research area, and focus, as well as any other information about yourself.
Thank you. Well, I come from New Zealand, and in New Zealand, most of our land area which is productive is in grasslands. And my work is involved in trying to protect those grasslands so they maintain productivity. But we also retain the soil carbon stocks, because we realize that's a very important component of the productivity.
Okay, that sounds interesting. And so within this project, what is your focus within the whole arouNd EJP SoilsC project?
So our focus is in terms of the different land uses associated with grasslands and different agricultural practices. Which of those practices helps us to retain soil carbon? In New Zealand, we have high soil carbon stocks, particularly in relation to Europe. Europe, our stocks are really very high. It's really important that we maintain those carbon stocks and prevent the loss of carbon. So we've been looking at different land uses and the impacts of those land uses on carbon stocks. And we've been doing comparisons between horticulture, between sheep and beef farming on flatland, sheep and beef farming on hill country, dairy farming, and cropping.
This looks like it's quite a lot of work. And from that work, what are the milestones that you have achieved so far?
we've been working with these soil samples from these different land uses. We chose 30 different soils in a region of New Zealand where the climate is very, very similar and the soil is the same type. So we were truly looking at the land use effect. There are big differences in the amount of carbon in soil under these different land uses. There are major differences in terms of the potential to add more carbon into those soils. And we've also detected differences in the vulnerability of that carbon to be lost.
And in all this work, have you faced any challenges or major discoveries?
Well, the major discoveries we've made is to be able to help us define a framework of vulnerability that we can then apply and we can take to our policy interests, people interested in policy, to help to say which areas are likely to be more vulnerable than others, which land use is likely to be more vulnerable than others. Previously, we've had a lot of information about soil type and in land use, but we've never had an index of vulnerability which we could apply at large regional scales.
And challenges, any challenges that you faced?
Well, the challenges as always working with soils is the variability in soils and being able to define the variability across a range of conditions to be able to synthesize what's actually happening. So we do need to take a large number of samples, and we do need to take samples across a wide region to come to some general conclusions.
And then any plans for the way forward from now?
Well, having done this for a small area of Canterbury, the area where I live in New Zealand, our challenge is to scale this nationally so that we can have an index of vulnerability nationally. And we are using different techniques to be able to assess the properties of the soils and their vulnerability using infrared techniques, cheaper cost options of measuring soil carbon concentrations and doing measurements more rapidly to be able to do that. And we believe that within a short time, we will be able to have a whole map of the vulnerability, and we will be able to inform policymakers about how they should be changing land use, or rather not changing land use to protect those soil carbon stocks.
I'm sure there's a lot of interest from the public in knowledge about vulnerability. How have you been sharing such knowledge with stakeholders in New Zealand?
Public is interested, yes. Farmers are interested, yes. They realise the importance of soil carbon. However, they also need to be offered incentives in order to make land use decisions which they would otherwise not make. So there's a lot of discussion in New Zealand about how we incentivise farmers in order to adopt practices which are going to make the land more sustainable. We work through our government and our local regional councils, and they set the standards and the requirements that farmers are required to adopt in their practices.
Okay, David, thank you so much for sharing your insights, and hopefully you will continue sharing, because I think there's interest from what I've gathered on this topic of vulnerability of carbon stocks. Thanks so much once again.
Thank you for asking us. Thank you.
So now I'm joined by Abad from France. Thank you so much for joining us. All right, you may please provide a brief background about yourself, your work, and your interest.
Okay, so my name is Abad Chabri. I'm a research director at the INRA Institute. It's a National Institute for Research, Agronomic and Environment and Food Security. And on the meantime, also I'm a professor at the University of Saclay, teaching biogeochemistry. Before that, I was spending almost 10 years in Germany before reaching France in 2003. So it is, I'm basically working, my background working on soil, soil sustainability, agriculture sustainability in the context of land use and climate change.
Thank you. Then, so within this project, what is your main focus?
Main focus is just I'm helping Melissa first of all to coordinate this project as it was in the beginning, trying to bring also my small experience in improving this project, but also trying to have the best what we can provide for the funders. Of course, what I'm expecting, because it is a very challenging project, not only in terms of thematic, but also it is a cross-continent, also dealing with the countries that in which we have less information and less data, specifically what's going on in Ghana, in Namibia, and all of this. And then it's an added value from what we have so far, but also it's an added value in terms of young scientist training and PhDs and all of these things. So this is really my focus, and I hope that we, at the end of this project, we come up with some strong results. Yeah.
You've also highlighted some of the challenges that you're facing. That was going to be my next question. Can you maybe elaborate more on the challenges of managing such a big project and how you've been coping?
Yeah, as usual, when you are dealing with different partners, different countries, and of course, when you are also trying to build up from what is already exists, is the first challenge is what data has been produced in the past, how we can collect this data, how we make sure that this data is available, and how make sure also this accuracy of this data, because it is a standing point from what we are going to build and from where we're going to develop further in this project. This is the first challenge. Second challenge is this, of course, you plan things in the project that never happen as you plan. You have all difficulties, not only natural difficulties, pedoclimatic difficulties, but also human resources, and then also it never work from the first plan. So you have to adjust, you have to adapt, and as I said, especially where we're working in different countries. But we have to keep mind positive, and then things is going.
Yeah, that's true. We need to keep pushing and be positive.
Yeah, absolutely. Yeah.
So what milestones have you achieved so far?
What we have achieved so far? And many things. Yeah, I mean, when speaking, generally speaking, at the French level, really, it was a very good opportunity because we are working on a long-term experiment. It is now started in 2005. And now we are in 2025. We have done what we call initial characterization of the system to see how much carbon we have in the beginning before implementing the experiment. And then one of the questions that we implement, at what extent the introduction of grassland within crop rotation, maize, wheat, barley, can or not help improve in the soil. So we have been, we know how much carbon we have. We know now how much is added or losing in the carbon. So we have been then doing this assessment in 2005, 2014, and then with the help of a PhD student from Brazil, we are trying to assess what we have in terms of carbon based on the management we implement. And what is very important, we see that some of the unexpected result that's difficult to convince a farmer is that we have some, how we call, information from the beginning that the grazing is increasing carbon in the system because uridang, labile compound, all of this, and it is not the case. And then the grazing is decreasing carbon in the system from what we seen from our result. But doesn't mean that these things is happening globally. Probably is a site specific, but it is a fact that we are observing based on the data that we are collecting. I think that's just to highlight, if maybe it will be in your question, and what's my expectation in the project. It is not just that every country work on its own corner and then trying to analyze, but try to have one, how we call, synergistic view in which how we can pull the data together in a way we can develop some of the questions that David highlighted a few minutes later about the vulnerability. For example, from what I heard this morning from his presentation, is that would it be possible to have the same approach? And I think the same is the case, same approach in a way that we can compare this approach with the data we have in France, but data we have in Ghana from Joshua, PhD, and then try to come up with a paper to highlight at what extent the soil and the particular management is responding differently, and what make the soil respond in that case. It is something that we can we can assess the vulnerability of the system based on intensification or disturbance regimes. This is some, and it will show it is a project that we are really, that was the expectation of the project, that we are working collectively, hand to hand, and we're pooling the resources and the expertise instead of just every country show his own result. Of course, this is a work package dealing with the modeling in which we are going to have all of this data to run the model, to test the scenario, and this is another question, but at least we can show methodological that's something differently beside the modeling that we are foreseeing in this project.
And I'm guessing one of the most important steps for that to happen is harmonization of the data. How have you been able to achieve that harmonization of the data for modeling, or something that is still ongoing?
You mean, yeah, this is, as I said, you mean if the data is available? What?
Harmonization of the data. That harmonization of the data for modeling.
The harmonization. Yeah, this is what Melissa mentioned this morning, that it's, of course, this is, we are trying to collect this data, put it on a different server. This is now, it is located in Brazil. But this is not that much easy, because once you have to collect this data, then you have to check this data if it is complete, if it is not complete, and then of course make it in a format that can be used for the modeling exercise. And I think that we hope that the extension of the will take place, because this is really what we need. We are reaching the third year in May of the project, and it will be a pity not to really to have this extension in a way we can test this data and run the model, and specifically some of the scenario that we promised in the beginning of this project.
And lastly, how have you been engaging stakeholders and farmers and students in France?
Yeah, I present some of the results, but I said they have some in mind, some of the practices that's relevant. Like as I said in the beginning, for them, grazing is increasing carbon. So when it comes, because it's a huge lobby in France for respect to grazing. Animals play very important, and I have nothing against, but also we have to see the animals also as disturbance regime. It depends how you intensify the system, but also with some consequences, of course, in terms of losing carbon, in terms of greenhouse gas emission. But it is a question of trade-off. We are not going to provide any magic recipe that can, in the meantime, reduce greenhouse gas and preserve carbon, but also provide food security. So this is a question, what are the expectations? Where we are going to put the cursor? We need like a high, in the middle, and then with the fact accepting the side effect, which is, we know that can be not 100% filling all of the boxes, but we have to keep in mind, and we have to accept some of the negative effect of these things. And it's sometimes for the farmer difficult to accept, and then it is a process of convincing, a process of making sure that the right way is this, but also we can reduce a little bit, but if we change altogether our mind of thinking, but our the not responding all the time to the market, and business. Yeah.
Well, thanks so much for sharing your insights. That was quite
thank you very much too. Thank you.
Okay, now I'm now joined by Joanna. Thanks so much for joining us. And my first question is to request you to give us a brief background about yourself, what you're doing, your studies, or anything that you can share on your background.
Yeah, hello. My name is Johanna Zimmermann, and I'm a PhD student in arouNd. So I live and work at the RPTU in Germany, but the experiment where I am working on is in Norway. It's a collaboration with the Norwegian Center of Organic Agriculture.
Thank you. So what is your focus within that project?
At our experiment, we are comparing the effects of comparing the effects of anaerobic digested slurry and untreated slurry mainly on the processes that happen in the soil. So my main focus is to identify processes that lead to a reduction of soil organic carbon, because most of those fields have now a decrease in soil organic carbon. We would like maybe to stop that or even increase this carbon stocks again.
And so far, what major milestones have you achieved?
So last year we did some soil sampling, and we also had greenhouse gas emissions. So we can say that the results indicate that when you apply the anaerobic digested slurry, even though it had less carbon in it, the carbon in it is more stable, so that could lead to a higher soil organic carbon content in the soil. So that's for once, but we also observed higher carbon dioxide emissions from these plots, but less nitrous oxide emissions.
And I know mostly in research we encounter too many problems and challenges. Have you encountered any challenges or problems in your research?
So my personal main problem is that I live in Germany, but I have to work in Norway. But I'm not allowed to be in Norway for a long time and spend a lot of money because of their foundation. So what I do, I drive there, take soil samples, take greenhouse gas emissions, and then I come back to Germany and do the lab analysis in Germany. So, for example, in the beginning of October this year, I was in Norway and I took 200 kilograms of soil with me in the car to do a column experiment during the winter in Germany.
Yeah, that seems like quite some challenge. So, any plans for the future going forward? What are your plans?
So right now we are planning, like I said, a column experiment during the winter where we would like to compare the two types of slurry that I mentioned earlier, and we also would like to see how the climate change will affect the soil organic carbon in soil. So we are also applying two different precipitation scenarios, so one with the historical data and one modeled future scenario where we have more precipitation to see how the processes are affected by climate change.
And lastly, have you embarked on any knowledge sharing events with stakeholders, with farmers?
Yeah, we hope to suggest something to the farmers in Norway because our research is really site-specific. But yeah, we hope that with the results we are able to say that maybe anaerobic digested slurry is better for the soil organic carbon.
Thank you so much for sharing your insights and interesting challenges. Hopefully, things will work out well for you. Thank you.
Thank you for having me.
I'm now joined by Professor Vincent Loga, who is one of our hosts. Thank you so much for joining us and for arranging this wonderful workshop.
All right, thank you very much. I hope you are enjoying your stay in Ghana.
Yes, it's quite interesting. Very nice campus here.
Great, great.
Okay, Prof. Vincent, my first question to you is, what is your focus within the arouNd project?
Okay, the focus of our work is to look at persistence of soil carbon in anthropogenic forest islands, or what we call the African dark earth. So we do quantify how much carbon is in the persistent pool, and then look at a lot of soil physical properties as well.
All right, thank you. And so far, what milestones have you achieved?
We are also working on a Norwegian site in a long-term experiment in Nibio. So we also looking at the same parameters in these areas. We have achieved a number of deliverables. We are training a lot of students, and at the same time linking up with farmers on how to manage their soils. And we have a number of stakeholders involved.
And within all this work, have we encountered any challenges or discoveries? Maybe let's start with challenge.
Field work is full of a lot of challenges, especially when you're handling a lot of data. I remember one of our sites in Ghana, we took over 200 samples. So we have over 1,000 samples taken across these areas. And that is really, really a lot of work. And then planning a lot of field work, and then also doing a lot of community entering, trying to, have permission to enter farmers' field. It's a lot of work.
And now going forward, what are the plans from now onwards?
For now, our immediate plan is to graduate our students, the PhDs to graduate, and then the masters to graduate. And then we also do much more of stakeholder engagement to disseminate our research findings.
Yeah, that's going to be my next question on how do we plan to engage the stakeholders and share information.
Yeah, for the stakeholders, we started engaging them along the project. For example, we did a training for farmers where we tried to train them how to prepare compost using recalcitrant materials to increase their persistent carbon pool and at the same time the particulate pool. And then we hope to do that more if we have the time and the resources.
Thank you so much for sharing those wonderful insights. And
all right, thank you very much. Thank you.
I'm now joined by Alan Manyevere from Forte, South Africa. Alan, welcome.
Thank you.
Okay, Alan, can you maybe provide a brief background about yourself, what you do, and where you come from?
Okay, my name is Alan Manyevere. I'm an associate professor of soil science at the University of Forte, and in this project, I'm a partner with Agricultural Research Council of South Africa. So basically, I can say, in short, that's who I am related to this project.
So can you share more details on your focus within this project?
Okay, so within this project, we are looking at improving soil organic carbon. Our trials are situated in an area where there is very low organic carbon. Some would call them dead soils, less than 2 mainly. So we have established CA trials or conservation agriculture trials 13 years back with the sole purpose of building organic carbon and also looking at soil and water conservation. Yes, so in this particular trial, we have three factors, the tillage systems, the rotation systems, and residue management. So for this project, we are trying to look at the persistence of soil organic carbon within the systems and also look at greenhouse gas emissions. Yeah.
Okay, thank you for that background. So what milestones have you achieved so far?
So far, I can say quite a number. So one of them is the recruitment of postgraduate students who have since graduated, two MScs, and we also have PhDs that we have incorporated within the project, and a research fellow as well who has been incorporated into the project. Yeah, in terms of the results, I think we have managed to see a gradual increase in terms of organic carbon. I think some of the results that we have presented show that up to more than 10% in terms of organic carbon. Remember, we are coming from a very low base, and that little 10% means a lot. So yeah, we have also managed to have a few publications and some manuscripts under review. Yeah.
Okay, thank you so much for those insights. So have you faced any challenges during your research work?
Oh yes, challenges. Most recently, we have had fire destroying some of our laboratories. In fact, the whole floor of laboratories has been taken out by fire, and unfortunately for this project, we have lost quite a number of samples for one of our PhD students who is working on soil microbiomes. So yeah, that has been a major, in my view, one of the major challenges. Yeah, that has been a major challenge for me. Yeah.
Any plans to recover from this for the student?
Oh yes, we are putting in place some measures. I think it's obvious that the student will need an extra year just to cover up for the samples that have been lost, and yeah, we intend to also just like, I think the extension of the project will also assist us in that regard, so that we can achieve that particular objective.
Okay, so one important aspect of this project is engagement with stakeholders and sharing knowledge with farmers and other stakeholders. Have we embarked on any of these previously?
Oh yes, sharing knowledge. We have shared knowledge at different platforms, presentations at scientific conferences, and also our students have been engaging with farmers around the project areas. Yeah, trying to sensitize them on CA systems and their benefits to the communities. Yeah, thank you.
Okay, thank you so much, Alan, for sharing those insights, and all the best as you go forward with your research work.
Thank you.
Thank you so much.
Thank you.
Okay, so as we wrap up, we've heard from various partners on what they're doing, their progress, their challenges, and their plans for the future. And we want to thank you so much for tuning in, and we'll keep on collaborating and working together. Thank you.
One quick question. When will the podcast be live, and where will it be shared?
Yeah. Most of today's podcast will be on all the see around social media, which Melissa had created. So we'll share almost all the link by the end of the workshop. But remember too that our national media like Joy News, Adom, and City FM are already picking some of these stories and already publishing them. So by the end of today, I'll be sending you links to what you did yesterday. Hi, Mr. Are you with us?
Yes, I was listening the interview.
Yeah, we can't hear you.
No? My microphone is opened. No?
They are going
Hearing
to work
me?
on. They are going to work on the mic for you. So
Mine?
we
Yes,
can't hear you. So she's going to work. They are work. The control room is working on your mic.
Ah, okay. Okay, it's better? I will test, 1, 2, test. No? No?
I think now we can hear you.
Yes?
Yes.
Can you hear me? Okay, thank you. The lady was talking, was Melissa, Ghanaian Melissa. Yes. Okay, thank you very much. I was listening the interviews. It's very, very exciting. I like it. Um, yes, we can share in our social media, and I'm trying to use to. We don't have yet YouTube, but I think I can create it together with our project account. So I think we can do it. Oh, I saw Jeroen in the room. He arrived. Is he arrived? Yes, I can see you. Did you see? Yes, it's me here. Like that. Yes. Oh, okay. Now, uh, welcome, Jiro. Now I think, I don't know, now you are in the lunchtime, I think. For me, it's in the middle of the morning, but for you, it's a lunchtime, I think. Vincent, would you like to speak?
Hello, Melissa.
Yes,
We have
I can.
Jeron just arriving from Germany. He's here with us at the moment. Jeron Zeto from Germany.
Yes, yes,
So
I
just
saw you.
introduce him
I saw him.
to the group that he's arrived safe in Ghana.
Oh, great. Thanks.
Okay. All right. So Jeron, welcome.
Oh, great. He would like to speak something about the update of the project.
Is it working?
Yes, the microphone is working.
Yeah, thank you for the welcome. Yeah, as said, just arrived, so getting climatized, so to say, because yeah, Germany it's now already getting winter, so a little bit different temperature. Yeah, but I'm looking forward to the discussions here this week. So, thank you.
Oh, great. Are you coming from Germany, being like, how do I say, that vegetable that we are onion. We are taking off the clothes in the in the way. So back into the podcast. I think after we can share these by our social media, and at the same time, I will try to put it on YouTube, because there I think we have a way to do it. But yes, at least from social media, and each a picture or anything else, what you providing on social media, please put our dot there from see around, and then we can repost and we share your information. And from now, Caleb, the program now, I think you are just a little bit late for the lunchtime. Yes, let me see the program. Yes, according my version of the program, I think that's it. Am I your microphone? Let me see if I. No, I can't hear you.
Hello.
Yes.
Oh, okay. I think
Thank you.
we are now up for lunch for about 40 minutes, and then we come for the lecture and the demo from selected participants from Ghana.
Oh, great, great. 1:30. Okay.
I hope you'll be having yours online.
Yes, I will. I will be here. Thank you very much for you all. Have a great lunch. See you soon. Thank you.
Hello. We are now on the lecture and demonstration section, where we will be looking at regenerative practices on its impact and innovations on soil carbon and greenhouse gases. We are supposed to have started with Dr. Alfred Atta, but he had some emergency in Accra, so could not make it to this particular section. But it is still very interesting that we are going to have Dr. Kofi Kunedu Boaten. He is coming from a specialized area in the sense that he was with us, but now he is with the founders. So we want to get both perspectives as he presents about a 20-minute word for us.
Okay.
No, not really. Just I have to make some notes about just to introduce the session. It's okay. I can. I hope that you are not sleeping after this heavy lunch. You need a coffee maybe. It works?
The one, the standing one.
This one? Okay, I said, yeah, good afternoon. So I have been asked to chair this session, but before we started, at least can give you some of the highlights, what we are talking about, and then why it is important, and then just to set the scene And yeah, in order to animate the discussion. So first of all, I would really highlight that, of course, when you're talking about the regenerative agriculture, we have to keep in mind that soil represents one of the largest active carbon pool on land, and that means those improving soil carbon stock offer a significant mitigation pathway. This is one thing, this was important things. And the second thing that we have to keep in mind also the term of regenerative agriculture, so what we are talking about, and it is often referred or used to describe a farming system that goes beyond beyond the sustainability, aiming not just to maintain, but also to restore soil biodiversity, ecosystem function, and carbon. So this is one we use about the regenerative in a way that we are having a kind of win-win situation, gaining and taking profit from the system, but also in the meantime, providing a kind of system that can be continuous and for the negative, but also for the positive way. So many agricultural soil, or as you know, not only in European countries are depleted in organic matter. In Europe, we have basically 1%, and then compared from what they have in New Zealand this morning, I heard that they are losing carbon, but they are far away from losing all the carbon that we have already lost in Europe. So it is, and then of course in Africa, but also in North Africa, due to many of these problems. First of all, the climate change itself and the erosion, but also misleading practices. So depleting organic carbon, as I said, tillage, crop residue removal, intensive practices, but also climate change. So that means if we are aiming to restore the soil organic carbon just for two reasons. The first one is to improve soil health. It's another concept that soil health, that the system aiming to provide the services, resilience to drought and flood, and of course nutrient cycling and also carbon sequestration to remove greenhouse gas emission from the system. So regenerative agriculture or practices may also reduce greenhouse gas emission. As I said yesterday and this morning, there will be or there will never be a magic recipe and a way that you have a perfect system, especially in agriculture, because we need food, we need to ensure the food security, but it is, I think that is it is now almost accepted by many countries, including the United Nations, that the only system continue to increase or to provide or to release greenhouse gas emission is agriculture. So we have to have a kind of compensated system from other part of the system that can also reduce or compensate from what we are losing from the system. That means we have to improve the soil management, we have to reduce the disturbance, we have to restore the nutrient use, but also the magnetism and performance and trade-off need to careful evaluation. So we need to have an assessment and evaluation, but also in a way that we can adjust the system. But it doesn't mean, and I have to repeat myself, the magic recipe will never exist in agriculture as far we want to ensure the food security and also reduce greenhouse gas emission. So I think that I'm not going to take so much time, so we have to show some of the examples of regenerative agriculture practices for soil and carbon greenhouse gas emission. There is many of the examples over the world, and if it works in one place doesn't mean it works in other place, because as I said yesterday, is a site-specific, region-specific, and then we have to adapt your system based from what you have in terms of. So this is many of the word that is been used to fulfill the regenerative agriculture. And for example, many of the use what we call the cover cropping, multi-species cover cropping, or at least introducing grassland into crop rotation within the different time scale in a way that we can, what we lose in agriculture, we can compensate in bringing this carbon again into the system. And it is not always very easy. Losing carbon is very easy. Bringing carbon to the system, it is another part of the business that needs always very forceful, but anyway, it is part of it. So we need to reduce the tillage. Many places the tillage may bring some of the added value and some positive, but not always the case. And then, for example, it works maybe in tropical area in Brazil, that's reduced tillage may bring some fruit and some, but it would never work. And in Europe, for example, such a practices has been abandoned because it doesn't improve carbon stock, and then even it was given some not the exact information of carbon stock assessment because you build this a very tiny area in which you compact the soil because the soil is not completely plowed, and then you develop, you concentrate carbon only in a few centimeters. It doesn't give you the real value on how much carbon. So it is one of the practices that has been abandoned, particularly or no tile or reduced tile. Basically, that now not 30 centimeters, but between 20 centimeters and 25 centimeters, but it's still almost plowing system is going on. The crop rotation and increased carbon diversity, crop diversity, it is also one of the practices that has been postponed by European Commission. Now we are not anymore allowed, since for 3-4 years it was in the former BARSO, it is not allowed to exist. I mean, after harvesting, you leave the crops, the system for 3-4 months until the next crop, and then by doing things, you lose a lot of carbon. But now such a practice is not. We need to have the soil completely covered with some of the kind of vegetation in a way that we can reduce. And most importantly, it was also stressed by the European Union, but also many, France in particular, but I think in Germany, what we call the crop diversification or multi-species. In fact, you can have grassland, you can have also legume, but also gramínea and mesa. It poses some of the problems in terms of harvesting, but also in terms of pest treatment. But it is a way how you can also improve the system in a way that, of course, you reduce also the amount of nutrition if you mix the legume and and gramínea. You have the nitrogen transfer, but also you improve the quality of the organic matter, which is also has an effect, as I said yesterday, in terms of greenhouse gas emission, because you may have reduced the nitrogen oxide emission, but also the fluxes in the system, but also increase the biodiversity, microbial composition, but also earthborne management. So I have been talking about the integration of crops and livestock, but also the trees and shrubs. It depends, and we use in many places integration of crop and grassland into crop rotation, but also in some other places they use shrub, but also integration, for example, in Brazil and some other places they use this thing. But of course, the integration of livestock and cropping system, grazing and the new plow, it not always was successful because we have had this experience in our long-term experiment when we plowed the grazing system for 6 years, we lost a huge amount of carbon because due to the label compound we have in the system, but also in the system itself, when you put animals in the system for the six years, we don't never increase carbon. Instead, we're losing carbon because animal, they add a label compound and stimulate microbial decomposition and then releasing carbon in the system. So you see, sometimes you have to test. It works in some places, but not in every place. And last but not least, of course, we have to improve, or we have to apply the innovative amendment versus soil carbon enhancement technology in a way that we can we can increase the amount of carbon by using, for example, biochar. But it also has been improved in many places, but not always very successful. It works in places, but not. So we have to adjust your system, enhance the weathering by, for example, by applying across silicate rocks, or to improve the system, or to salt to accelerate chemical uptake of CO2, because it is silicate has been also used in many places as a carbon sink to absorb CO2. But also we need to, and then I think we've been yesterday talking about the in the round discussion about the AI, the artificial intelligence, that really can be used in this case, not for generating papers, but also for or calculating, but also applying to tell you what is the best way, how you can measure the evolution of carbon stock, but also monitoring, improving the monitoring, and also improving the report and verification, MRMV, and of carbon changes. And thus enabling scaling the quantification of regenerative practices. Another problem, and here that when we talk about the practices, if it works, this is always what we practice in Europe, but also in agriculture. It works when in one place you get a very successful, you say, wow, it works, we are happy, but when it comes to upscale, this practice is another problem. You have to have the all ingredient in a way to upscale and make sure that what you have observed in your area, in your region, is scalable, and also make it successful. So this is some of the keywords I wanted to share with you in a way that we can stimulate some of discussion. I am sure that it is not complete, and probably you have other information and other idea on how we together we can come up with some of the solution in a way that we can put it into practice. Thank you.
Thank you very much, Abad. It will be interesting for you to share your slide with us. We like the point.
With you. It is just because Johanna told me you have to jump. I said, okay, I have to leave my lunch and jump.
Yeah. So, Abad, your chair is there for sure. Oh, okay. So, our next presenter will be Dr. Frank Aidan.
Good afternoon. My name is Frank Aidan. I'm from the Department of Animal Science, K University. This afternoon, I'm trying to see if I can say something on the greenhouse gas emissions. Looks like everyone here is a soil or crop man. and I'm the only animal here. Yes. So if the crops are not there, the animals we are in trouble. So if you are doing your things, please consider us because we need your crops to survive. All right. So I'll be looking at some few practices that can help to reduce greenhouse gas emission. So this will be my outline of the presentation. I'll go straight to look at some few points, and then I come to look at the innovations and then the practices that can help to reduce the greenhouse gas emissions. Now, the population of the world is growing very fast. As a result, we need to provide food for the individuals who are growing, who are being given birth to every day. It's estimated to have about 9 billion of people in the next few years. All these people will have to eat. And food is not only plant. And it means that we need to try and then supply animal protein, especially in the developing countries. Once the middle class is growing and the incomes are growing, there's dietary shifts and people are demanding more of animal protein compared to plant protein. So because of this, there is a lot of intensification of farming, and that is one of the major reasons why we are having issues with the greenhouse gas emissions from livestock. So this growth that we are talking about has resulted in higher land use and water use and increased greenhouse gas emissions. Here we are looking at gas like carbon dioxide, nitrous oxide, and methane, with methane being the most potent of these gases. So the impact of the livestock growth will be on soil health, deforestation, global warming, air quality, water pollution, and environmental sustainability. So if you take soil health, for instance, you want to keep animals on pasture, and then as they graze, they can cause overgrazing, which can result in the loss of the topsoil through erosion. So the major greenhouse gases that are of importance have been estimated by different agencies, with our Intergovernmental Panel on Climate Change, FAO, EPA, and all the others, estimating these emissions from livestock to be between 8 to 18%. So that is quite a lot. So out of these gases that I mentioned, methane is the most potent of them, and it has a lot of concerns when we talk about greenhouse gas emissions. And it is estimated to be more than 25 times that of carbon dioxide. And this is due to the fact that there are different animals which contributes to methane production. But the livestock that of importance are the ruminants, with cattle being the most animal with the highest emissions of methane. So the direct methane that are generated by cattle or other ruminants are from two sources. The first source is enteric fermentation. So during the digestion process, there is fermentation, and this results in the production of methane. And this is normally due to the fact that if the feed is of poor quality, you are going to have a lot of the fermentation products being converted to methane instead of being shifted into propionate, which can be used as energy source for the animal. The other is also on manure management. So the way we manage our manure can also result in the release of methane into the atmosphere. So for enteric fermentation, if we look at ruminants, unlike monogastrics, they have a rumen which is capable of breaking down or fermenting fibrous materials. And as a result of this fermentation, there are some microorganisms within the rumen which we call the methanogens. And these are able to convert hydrogen and carbon dioxide into methane. So the more you have methanogens in the rumen, the more you are likely to produce more methane from the animals. And once the animals belch or defecate or release any gases, this methane can be released into the atmosphere. One of the major problems will be for animals that are grazing, especially those that are moved from one place to another place on poor quality roughage. And these animals hardly receive any concentrate or highly digestible materials, and that results in production of more methanes. So the production system is one factor that affects methane production. If animals are housed or in the intensive system and provided with concentrate or highly digestible roughages or forages, the amount of gases released in terms of methane is quite low compared to animals that are grazing on poor quality roughages. But in terms of production per animal, the dairy cattle, because they consume a lot of feed, they have higher methane output. But for beef cattle, which are normally grazed on poor quality, per feed consumed, they release more methane compared to daily cattle. The other way that can also contribute to the release of methane is how manure is managed. How manure is managed. And methane from manure is normally as a result of anaerobic decomposition of manure. And these are normally manure that are stored in lagoons or slurry tanks or pits. If they are exposed to anaerobic decomposition, then you will likely to have the release of methane. But most often, if they are allowed to be decomposed using the composting procedures, then they are shifted from methane to carbon dioxide and then nitrous oxide, which is quite manageable compared to the methane. So in terms of nitrous oxide, it is the third gas that is of concern compared to methane and then carbon dioxide. Now, as the concentration of the nitrous oxide increase in the atmosphere, it can affect the stratosphere, and then that can result in imbalances within the radiation on Earth. So research has indicated that the presence of nitrous oxide in the atmosphere can result in formation of acid rains. And that is one of the things that we want to avoid. But for nitrous oxide, even though the effect is not too sudden compared to methane, it has a longer span in terms of its storage in the atmosphere. So if we allow the gradual buildup of these gases in the atmosphere, they can cause long-term problems for us. So mostly the nitrous oxide released from animals are from manure management. If manure is improperly managed, for example, during nitrification, it can result in the release of nitrates, and then during denitrification, the anaerobic microbial conversion of nitrate into nitrogen gas can result in nitrous oxide, which is an intermediate of the process. Even though this is smaller compared to that of fossil fuels, as I said, if they are allowed to accumulate over a long period of time, they will be of a challenge to us. So we have to try to avoid the continuous release of these gases into the atmosphere, which might eventually cause long-term problem with the climate change. So for nitrous oxide, if the manures are not managed properly, and then there's nitrification, it's going to result in the release of these gases into the atmosphere. Apart from these direct sources, there are also indirect ways through which methane and nitrous oxide are emitted into the atmosphere. So for methane, indirect causes normally result from decomposition outside a controlled storage systems, even though it is not as much as that of the direct methane emissions. They are also very important, and they can result in overall increase in the methane footprint. So here, the key pathway that we have to look at is to how to manage manure. And here we are looking at how manure is what is applied onto the soil, because improper application can lead to the release of nitrous oxide. So when you have waterlogged areas, when you have soils that are that contains a lot of water, and these manure find their way in there, because of the anaerobic nature of the place, they can result in release of these gases. So there are methods of estimating these gases. It's quite complex, but I'll try to just give a brief overview of that. So for methane and nitrous oxide emission estimation is quite a complex thing. So there are different ways that we can try to estimate these gases. But before you can do that, you need detailed data on the specific type of animals that you are using, including even the diets that they consume. So you have to compute for feed intake. You have to check the digestibility of the feed. So here, if you are looking at the feed composition, you have to look at the protein content of the feed, and then how digestible this feed are. Usually, diets that are highly fibrous are difficult to digest, and that is what usually creates the fermentation issues that result in the release of methane. So the management system is very important, whether they are on the extensive system or intensive system, as well as the local climatic conditions. So for methane, there are usually two pathways that they get into the atmosphere. One is emission. If you want to calculate, that would be an emission from the digestion process. So the enteric fermentation is one of the major problems when it comes to methane release from livestock. And then the other is what? The manure management. But for nitrous oxide, it's mostly as a result of how we manage manure. And in terms of this, we are looking at how manure is stored, whether they are liquid or they are solid. They determine the amount of nitrous oxide to be released. And then the application method that is used on the soil, whether we just dump the manure on the soil or we compose the manure before we use that to fertilize the soil. So in terms of estimation, we normally use the IPCC method. We have tier 1, tier 2, and tier 3. Okay, in Ghana here, we are trying to use a tier 2 method to estimate. The tier 1 is quite simple, but the tier 2 is something that takes a lot of effort. Because one, you need to know the subcategories of the animal. So even with cattle, you have to categorize them into what? Into beef cattle and then dairy cattle, or dual purpose. And if they are dairy cattle, you have to look at calves, you have to look at all the different ages. So the subcategorization of these animals is what puts the difficulty here. And you need to know the population of what? The cattle in the state. And you know, Ghana here, anytime you want to do census of any animals, we are going to face a lot of challenges because the farmers might think that you are going to use that to tax them. So they will, if they even volunteer to give you the data, they will underdeclare the number of animals. And you know here also, people think that when people know that they have so much animals, they may think they are rich, and then their responsibility will increase. So these are some of the things that make the estimation using the tier 2 a bit difficult. But nevertheless, we've been able to achieve a lot with this tier 2 method. So for the tier 1, as I said, it's very simple. You need to know the number of animals that you are using, and then you just try to get that by multiplying that with an emission factors, which are already established by IPCC. So you get the population numbers, and then you multiply it by that. So this method is mostly useful for initial estimates of greenhouse gases from livestock. And then they provide countries with a standardized and accessible way to estimate greenhouse gas emissions. So here you can do that for all countries. So it makes it easy. If you don't have specific data, you can just pick the already established data and then use to do that. And this can also cover both enteric fermentation and then manure management. So for the enteric fermentation emissions, they are normally estimated by multiplying the number of cattle by the global average of emissions. So using the emission factors, and that is what is going to help you to get there. But you have to know that the emission factors differ based on the type of animal, whether it's cattle, sheep, or goats, or the subcategorization of the animal. For manure management, emissions are calculated using the standard manure production values. And then, and these are then converted by using the factors that I mentioned about. But one thing about this procedure is that even though it can help you to compare between countries, it only uses basic livestock populations, and then it's try to underestimate how much emissions that are available within the sub categorization. So it is the simplicity that we are talking about here is based on the fact that it is standardized in terms of how it is reported. So you can pick values from one country and then use that emission factors to calculate for another country. So because of that, it is inaccurate in reporting specific conditions within different countries. So regionally, you may have issues. So even in Ghana here, there are different ecological zones. So if you are relying on the tier 1, you may consider both the Guinea savanna, the forest zone, the transitional zone as one. Meanwhile, the conditions over there are different, and that means that you have to consider that differently. So based on this limitation, then the tier 2 procedure was instituted. And this procedure tried to do that based on country specification. So every country is tasked to produce their own estimates for these gases. So for enteric fermentation emissions, this uses local data. So you can't just pick data from anywhere and use. You have to generate the data internally and use that. And it also have to consider the type of animal that you are using, whether it is raised intensively, whether it's raised extensively, whether it is for milk production or meat production, and then the feed intake, as well as the composition of the diet or feed that you are providing for the animals. So for manure, using the tier 2, you have to consider local storage systems and climate-specific methane conversion factors. So every specific area, you need to generate these conversion factors to properly estimate the methane emissions. And this is more flexible. So it gives us room to sometimes use expert judgment and so on and so forth to get the estimation done properly. So this procedure is more accurate compared to the tier 1, and it accounts for both extensive system and then intensive system. All the system of productions are accounted for. And because of the fact that it is country specific, it is more credible in terms of the data that is generated from there. So here It captures the emission reduction from improved practices. So for example, if this year, these emission factors that you got, and then farming practices have improved and conditions have improved, you have to go back and revise. It's not static that once you get these factors or this emission factor, then it is static and it's going to be forever. You have to review it over time once conditions improve. So because of that, you can create a tracking tool to be able to follow and then make adjustments as and when conditions improve. So it helps us to target greenhouse gas reduction by focusing on policy, because when we explain how these contribute to emissions, we can try to bring out practices that would reduce these emissions. Once we bring out proper practices that reduce emission, then we have to go back and then update the values that we have. So for the mitigation strategies, we are looking at using the regenerative practices. So in livestock production, when we talk about regenerative practices, it might not be the same as what happens in the soil or crop side. But here we are looking at addressing dual challenge of what? One, reducing greenhouse gas emission, and then enhancing carbon sequestration. Because if we are reducing greenhouse gas emission, we also have to try to improve the carbon storage so that the plants that the animals will consume also grow well, and then they can get good forage. So it has various combination of factors that it uses. It's approach livestock farming in multi-dimensional way. It looks at land management, manure handling, and then the use of what advanced innovations. So how do you manage your land? Are you using crop rotation? Okay, are you using land rotation? Whatever procedures that you are using, that conserves soil moisture. The idea, soil moisture, the idea is to limit the usage of synthetic materials as much as possible. So here we are looking at synthetic fertilizers and the synthetic chemicals or pesticides for controlling pests and other things. And when these are properly done, then to pave way for sustainable and climate resilient livestock sector. So here we, even though we are animal specialists, we are also looking at how the crops that are used to feed the animals are grown and whether they are conforming to carbon storage. So, with regenerative livestock farming, it can sequester carbon by improving soil health through practices like rotational grazing. So here we are looking at frequency of what? Of moving the animals. You don't want to leave the animals too long on what? On one paddock so that they graze to the extent of even eating the roots. So you want to maintain the plant as high as possible so that they can regenerate quickly for more pasture to be obtained for the animals. So this approach mimics what happens naturally. So when farmers send the animals to grazing, they don't stay at one point for a very long time. They move as and when the site better forage ahead. So you do that and then you minimize the use of fertilizers because once the animals are there for that short period, all their dungs can also be used to improve the soil fertility. So this cycle that we are talking about will allow the plant to regrow very quickly. And once they regrow quickly, it means that the roots will not be destroyed by the animals, and that will increase the soil organic matter. So the system here is such that it pulls carbon dioxide from the atmosphere and then stores that in the soil. Because the plants are still going to remain on the soil, and once they are there, they will trap carbon dioxide from the atmosphere and use that to convert or produce their food through photosynthesis. So this is a way of showing how this integrated system can help to improve soil fertility. So you have your your land over here. So you are preserving the roots, okay, of the perennial crop. So meaning that you don't want to keep the animals too long. Once the animals remain here for too long and all the greens are finished, they will try as much as possible to get food to eat, and then they will be pulling the plants, destroying the roots of the plant. Once the roots are destroyed, it means regeneration becomes difficult, and then you wouldn't be able to have enough mulch on the soil. So the practices that I talked about, I am just trying to put some few here. So we are looking at rotational grazing. Here, systematic movement of animals from one location to the other. So it is best done when they are in paddocks. So if you have a fenced area, then you have maybe 20 to 50 acres of land. You have grown your grasses and your legumes with your agroforestry plants in there. What happens is that you paddock them, you divide them into sections. So you move them from one section to the other. So by the time they move around and come, the first one is what is ready. So at any point in time, there will be enough forage for the animals. And then they won't put too much pressure on the plants to even think of removing the roots. Then manure management is also one thing. So how you manage manure in terms of storing them in facilities, managing the moisture content and oxygen. All these things helps to minimize the release of methane and then nitrous oxide. Then you can also have an integrated system where you combine livestock with crops. So here, the manure from the livestock will provide nutrient for the plants, while the animals will consume the plants. So the next one, which is of very important to we, the feed scientists, is the provision of improved feed. Because once the feed is improved, once the feed is highly digestible, it means that the amount of methanogens within the animal will reduce, and then we are going to shift the digestion products, that the volatile fatty acids, to propionate, butyrate, and acetate for the animals to use, instead of being converted to methane, which is what we are trying to avoid. So for the innovations, we are looking at feed additives. So we can provide feed additives that inhibit methane production. So we have methane inhibitors. So a typical example is a seaweed. If we supply seaweed, we can limit the amount of methane that is produced. So here, the rumen environment will be improved, and then the bacteria that ferment the product to release acetate, butyrate, and propionate will be higher than the methanogens from the archaea, which result in production of methane. Then also genetic selection. We can look at selecting animals that naturally will emit lower emissions of methane. So here you are targeting the animal and then improving it to a point where its digestive system will be able to handle products with minimum release of these gases that we are talking about. And this can be used to target long-term efficiency in livestock production, in reduction of greenhouse gases. Then also using the manure that is being generated to produce gases, okay, that's the biogas, to power the machines or whatever within the farm. So here you are using the energy, which otherwise would just be converted to methane and be dissipated into useful activities. So with the use of, this is a normal thing that goes through a typical cattle. So it takes in the feed, there's bacterial fermentation or digestion, and then it results in the production of volatile fatty acid, hydrogen, and carbon dioxide. So depending on how much of these materials are produced, if more hydrogen and carbon dioxide are produced, and then methanogens will then convert this into what? Into methane, and that is going to be a problem. But if you provide feed additives, these additives will limit the amount of carbon dioxide and hydrogen produced, pushing most of these into volatile fatty acids, and that will be useful to the animal, and then that will result in reduction in these methane and nitrous oxide. So that is what happens over here. So your target is to the rumen environment, improving it, and then ensuring that you have the right form of what of microbes to ferment the product for you to get your end product. So that is what we have here. So in conclusion, regenerative farming moves beyond simply reducing emissions to actively restoring ecosystems and sequestering carbon in the soil. So here, the target is not only about reduction, but making sure that you have enough carbon stored within the soil. So here, by integrating practices like crop rotation, as rotational cropping I talked about, or rotational grazing, you can improve how also you manage manure, and then with this integrated system, you can create a livestock system which is resilient and will result in reduction of these gases. So this approach that I'm talking about is powerful, and this can result in mitigating climate change while ensuring long-term viability and then sustainability of livestock. So I think for the take-home message, we are looking at regenerative livestock farming as a tool for a dual-purpose solution. And then this solution is one, reducing greenhouse gas emission and also trying to remove carbon from the atmosphere and then storing them within the soil. So by working with the natural systems here, you are not doing too much use of artificial materials, and that can help to transform livestock production. So thank you very much for your attention.
So summarizing your presentation, what would you say would be the best strategy to reduce methane from livestock?
So the best strategy would be to improve the feed that you offer the animal. Once there is improved digestibility, there is going to be reduction in methane production, and that can reduce the greenhouse gas emissions.
Okay, thank you. And did you any studies about this topic?
So for now, we are currently gathering data to produce the tier 2 greenhouse emission figures for Ghana. So we are almost done. But there have been studies that have been conducted within our lab. And then we have realized that if you provide little concentrate, that's our concentrate, we are looking at highly digestible, either for energy or for protein. So if you have some little amount of soybean meal, you have some little amount of rice bran or wheat bran. These are a bit easier to digest, and that improves the rumen function, and that results in a reduction in methane production. Mind you, in Ghana here, we have 2 major seasons. During the dry season, the forage availability is very, very difficult. So the animals rely on poor quality roughages. And that is where the issue comes. So if we train our farmers to at least provide some little form of supplementation, it will improve the rumen function, and then we can reduce the greenhouse gas emissions from the animals.
Okay, thank you.
Thank you.
Further question?
Hi. I am the next after.
Hello.
Hi.
Good presentation, Dr.
Thank you.
Aidan. You talk about the ruminants. I want to shift to the chicken, the poultry.
Yes.
Yes. Rearing the layer and that of the broiler, which I think converts much feed. Based on your analysis, I'm presupposing that rearing the broiler becomes more efficient in terms of climate emissions.
Yes, because for poultry, they are normally provided with easily digestible feed. Okay, so yeah, you're looking at maize, soybean meal. These are highly digestible nutrients. So in terms of excretion, and they also do not have the rumen where microbial fermentation Okay,
zones, yeah.
okay. Only small amount of fermentation takes place within the large intestine. It's not enough to cause so much problem. So in terms of efficiency and in terms of reduction of greenhouse gases, monogastrics are much more efficient than ruminants.
Thank you.
Thank you.
Because within our sector, we use more of the poultry manure than the other manures. That's why I'm interested.
Yes, but it's better to do composting before you use so that you don't allow those gases to escape into the atmosphere.
Thank you.
Thank you.
Chair, Dr. Aidan, for a very nice presentation. I want to ask if, apart from feeding, another aspect of animal production, which other key areas do you think are very much culprit for emissions?
Okay, so I can say deforestation. For instance, if you want to grow pasture, you can't keep the original trees on the land. So you might have to remove these plants, and once you are removing these trees, automatically you are reducing the ability of the area to absorb carbon dioxide from the atmosphere. It can cause that. And then also how we manage the animals. So if we do not manage the animals properly, as we allow them to consume anything. So the management for animals, I think feeding is the key thing. But before you can get the feed, there are other distractions that we also do, as in land preparation for planting and all those things. And then maybe within the production unit, if there are tractors you are using, they can also emit a little. If you are processing pig processing facilities, how the waste released can also result in storage of these products. And then in those animals that are stored, raised intensively, also if you are using the slurry tanks, you are going to use liquid manure because you have to wash the rooms all the time with water. And these can create anaerobic conditions that can result in release of these gases.
So processing too is very, very key.
Yes, it's key.
Okay, thank you very much.
Further question? Edward?
Those of us coming from the cropland or the agricultural crop production side and the livestock sector,
yeah,
who is the major culprit in greenhouse gas emissions?
Honestly, so we have to look at the percentage contribution of greenhouse gases. Livestock do maximum 18%. So if we know what, how much crop does, we can easily know where the profit is coming from. And then I think for livestock, it is easy to manage if you understand their physiology. So once we know that improving the digestibility of the feed, improving the quality of the feed can result in reduction in that. If we do that, we can easily reduce emissions. Thank you.
I think
looks like the questions are not finishing. Okay. Yes, sir. Yes. Yeah, we can improve pasture, as you said. But one thing about pasture is that if they are also mainly grass-based, they still have a problem with methane emissions. You want more legumes than just grasses. So it will be a multifaceted approach if we are able to combine legumes with what? With grasses. But anytime we talk about pasture development, it is always about grass. But there are Salazantes, Cajanus, all these plants, they have high protein content. And once there's high protein content, the fermentation favors the bacteria, and the bacteria is able to convert most of these products into volatile fertilizer for the animal to use. So we can improve that, but you see, pasture development is very expensive. Imagine you have 1,000 cattle, and you want to develop pasture for the cattle. you may need not less than 20 hectares of land if you want to proper sustainable farming. And to establish this thing without irrigation, in our case where we have to rely on rainfall, it's quite difficult. So the farmers want the easy way. I won't say the easy way out, but it's more or less like the only option that they have. So if we are talking about concentrate, it does not mean that how you are going to provide it on like you are supplying the feed to them. It's just a supplement. So you are not going to go beyond 30%. So between 10 to 15%, just to improve on the protein intake. That will help the animal. And it's not as expensive as establishing quality pasture because the seeds, at least, how much is Brachiria? So 1 kilo is $100. You have 100 hectares of land. How many kilos can you buy? Yeah.
Sorry, could you please use the microphone?
Okay.
Here from audience, we can't hear.
Okay, okay, good. I'm saying I'm aware government some time ago facilitated the spraying of stylo seeds, but it appears it didn't work well. I don't know whether the animal scientists were not involved or something, because the stylo, they have a very robust variety which does not need much care. And there is also, let me use the word feeble or weak variety which does not survive. Well, I don't know the role you played in this thing for the failure of that exercise.
Yeah,
because,
okay.
please, farmers, even if it is 10%, I don't know the cost estimate you made. If it is not free, it will be very difficult for livestock farmers to afford concentrate. That's why they leave them even to chew plastics, and sometimes they even die. They are aware of the danger, but they're not able to come out of it. Thank you.
Yeah, thank you very much. I think for the stylo that you are talking about, probably the way they were sprayed, the seeds might have not touched the soil, because if it is not a prepared land, and then you just spray, they might end up falling on leaves or on things that means that they are not going to get access to the soil to germinate, and then grow as a result of that. I don't have too much information on that. But I think, as you are suggesting, if farmers can also do even 10% of pasture, it can help them to reduce their cost of production.
Aidan,
Yeah.
this one is just a small query to what global agencies are saying about animals and to solve the methane issue. What's your take? Let's stop eating animals. Let's stop eating ruminants and all, and go back to insects.
I don't think we can stop eating ruminants. I mean, we just have to raise them sustainably. Now we have information. Once we have information, we should be able to use that to produce them in a sustainable manner. Insects are very nutritious in terms of protein, but how many people will be willing to consume locusts, cockroaches, black soldier flies, and so on and so forth? I mean, once you have money, your taste changes. And I don't think you want to use your money to buy insects, even though people will easily consume them if it is packaged properly. But I think the most important is sustainability. If you do it properly, we wouldn't have too much of a problem.
Microphone.
I asked that question because I know you'll be going for global programs, and you'll be asked this question: why don't you go away with ruminants and then?
Yeah, I mean, ruminants are so precious to those who raise them that if you say we shouldn't do that, it will be a big problem. Because if you go to some part of Ghana, they don't even want to slaughter the animal. They just want to see the animals plenty on their field, and they are happy. It gives them a lot of prestige. So that's what it is.
Any further question?
So I would like to do a question. I don't know if we have a line.
Okay.
Just, may I?
Time is.
Okay. Here, I wrote in the chat.
Yeah, it seems, no, we have our three presenters.
Okay.
Oh, then.
Okay.
I think Melissa is saying something online.
Yeah, may I? Just a little bit. Just a little.
Yes.
Here.
Okay, sure.
Congratulations for.
All right.
Thank you very much.
Okay. I wanted to ask you some questions, but you can do it later on. But anyway. Okay. Thank you very much.
Thank you.
It's no time. Otherwise, I will be. So, who is the next speaker? Yeah, I didn't have the agenda, so I didn't take care about the time. Sorry. So when we should stop? At 3:30? Okay, then he explores the time there. Yeah. Okay, so we take item. 15 minutes.
All right. Thank you very much. Is it audible enough?
Yes, please go ahead.
It is. Okay.
So you have, did you have 15 minutes?
Yes.
10 minutes.
I'll try and
10 minute discussion.
I'll try and do it in a few minutes.
I will be sharp on the time.
Yes, yes. So thank you very much, and thank you to Caleb for inviting me here. So when Caleb said I should come and do this presentation, I asked him what he wanted me to say. Thank you. What he wanted me to say. So he said, well, there will be scientists here, and you work with a funding organization, so they would love to listen to you. Good. Nobody got the joke.
Yeah, no, it seems like your mic is not.
It's not. It's not. It's on, but this one works. Yeah. Okay. Thank you. Yeah. So my name is Kofi Boateng. I work with the Global Methane Hub program office, and I lead on our rights programming. So I will quickly, take you through what we do. And Dr. Aidan did quite a lot of work on what I'll be saying. So that's a good scene for me to continue. So the Global Methane Hub is a philanthropic organization, basically. So we focus on methane mitigation across 3 key sectors. So remember at Glasgow in 2020 COP26, there was a huge focus on methane mitigation. So our organization came along during that period to provide the impetus that we would need to bring action to the global methane pledge, which is the goal is to reduce methane emissions by 30% from 2020 levels by 2030. So basically, we focus on the energy, agriculture, and the waste sectors. And these 3 sectors alone account for 96% of the emissions from methane emissions from that emanate from human sources. So we collaborate with governments and non-governmental organizations to develop effective solutions and then look for the pathways that these solutions can be scaled for impact. So why methane? And Dr. Aidan provided a good context on why it's important for us to focus on methane. So the current warming that we experience, 45% of that is because of methane emissions. So that is really critical for us to look at. And methane also because it is highly potent greenhouse gas emissions. We know CO2 is the key focus, but in terms of their global warming potential, methane is 86 times more powerful than CO2. So to provide context, if carbon dioxide can put 1 hot coal in their pocket, methane can do 86 times of that. So that is really, really critically important for us to look at. But also the good news about that also is that methane doesn't stay in the atmosphere for long. Averagely between 10 to 12 years, it's gone, whereas CO2 stays for hundreds of years. So what that provides us, or the opportunity that provides us, is that if we are able to significantly reduce methane emissions, that surely is our best bet in reducing warming in the near term. So that is why it's important that we focus on methane emissions. So there are sectors that account for methane emissions globally. I'll try and use this. Good. So I'll come to the pie chart here, but before that, the agricultural sector is a very important sector when it comes to methane emissions. If you look at the livestock and then the rice subsectors, rice alone accounts for 8% of global methane emissions from anthropogenic sources. And if you look at fossil fuel combustion, so you could see the transport industry. If you compare that to rice alone, rice emits more methane than that. If you look at livestock, it's the largest single source of methane emission. And that is huge. And so the mitigation potential for these two sectors is really good if we have the solutions to be able to drive down methane emissions. But in agriculture, there are challenges that we face in terms of developing these solutions and then scaling them for impact. The solutions that are currently available, they are not cost effective, and then they don't have proven effectiveness for absolute reductions. So mostly it is the intensities that are reducing. For instance, in rice, if you produce more rice with the same emissions, methane emitted, then intensity is low, which is good. So just by also increasing the efficiency of production systems can be a mitigation strategy, which is what mostly is occurring now. And for farmers, and as he was saying, farmers need to be incentivized, and it is important that they see the benefit from the actions that they are going to take before they commit to it. And so currently the solutions that are available, very little productivity gain to the farmer. And so it is hard for them to adopt and scale. For those that provide some gain, I mean, the worst is to maintain the productivity levels and not to have them additional cost to their cost of production. And for agricultural solutions, so there's no plug-and-play options. So you can't pick one solution that worked in, say, Kumasi, and then the same solution can be effective even within a country, maybe in the north. That needs to be adapted to local dynamics of the area that you intend to apply these solutions. What we cannot measure, we can't manage, and it is basic. For agriculture sector, measuring or monitoring methane emissions is difficult, and especially so for livestock sector. The animals are moving. So how do you measure the methane emissions that they are producing? So it's very, very difficult. Even in the rice, subsector also, the most common method that is used is the chamber method, where you can't really apply it at scale. So it's within the field, and even that is not being done enough, especially in the global south. And so there are large uncertainties in the kind of the data that we have, the available data, and so we resort to emission factors, which do not really reflect the dynamics that exist in those production areas. Now come quickly to the pie chart here. If you look here for rice, so I'll focus more on the rice now. The solutions that are available are technically viable around 50%. What this means is that these solutions, yes, technically they can be used to reduce emissions, but really they are not being used because that's why they are technically viable. Those that are applied, the economically viable ones are at 6%, which is very small. Basically, these are water management practices, basically, because for methane to reproduce, there needs to be flooded soil, anaerobic environment for the processes to take place. So for us to be able to really get low emission outcomes, that will significantly reduce methane emissions from the rice sector, we need to increase the slice of economically viable mitigation solutions. And that's why what you're doing here is really, really important. The generation of solutions, innovative solutions that reflect the dynamics of production systems, especially in the global south. So at the Global Methane Hub, our goal is to support the reduction of methane from the agricultural sector by 10% by 2020 and then 50% by 2050. And we do this, or we are doing this through the following 6 strategic areas. Improvement in measurement and accountability is really important. that we're able to improve our monitoring and the reporting, and also the verification in both rice and livestock systems. And it's also important that in the development of these capabilities for improved measurement, they are low cost and they're robust. That can be applied at scale. It's really, really important. And then win-wins. Our approach that we support or we advocate for is to look at the opportunities within the production systems where the farmer can have a benefit in implementing or adopting the solutions that we generate. So win-wins are key, and they are the entryway or the pathways where we can have mitigation action done. And especially in the Global South, adaptation is key, and it is right, it's rightly so. But through adaptation, we can have mitigation outcomes also. And that's why we call it adaptation-led mitigation. For instance, so if the farmer, the rice farmer, for instance, reduces water usage on his field, what it does is that it saves him cost. It builds their resilience. And in so doing, methane emissions are also reduced because if there's little water on the field, the production is curtailed. So that is the approach that we emphasize and encourage in our programming. And also accelerating R&D. I said earlier that for agricultural mitigation solutions, it is not one type fits all. They need to be adapted to the environment which they have to be applied. So it is important that we accelerate research and development to be able to identify the opportunities within specific environments so that the solutions that are developed to be implemented there becomes effective, and then they are owned by the people or the farmers or the stakeholders within those environments. Encouraging company action. I think in reducing the impact of emissions on the warming planet, it cannot be done in isolation. And a key stakeholder in all this are the actors on the value chain. So for livestock, dairy companies, beef companies, for rice, millers, those who buy it in bulk for production to fast-moving consumer goods. These are all critical players that need to be on board in terms of finding solutions for methane mitigation at the agricultural subsectors. And especially for rice, farmers in the Global South, farmers are smallholder farmers, and a lot of the production that they do are bought off from them, and they're used in the production in big multinationals. And so if these multinationals commit to source rice that is sustainably produced, then in effect, they're also helping to propagate sustainable farming practices at the smallholder level. Then the influencing of public policy. We work with multilateral development banks like AFDB, Africa Development Bank, to make sure that they are mainstream methane in their lending portfolios. What this does is that it helps to increase the finance available for climate action in terms of methane mitigation. Currently, climate finance that is available for methane mitigation is less than 2%. So there's for impact, we need to scale. And scaling means finance must be available. So how do we catalyze agricultural methane mitigation action across the world? We do that through strategic convening and then effective grant making. And by strategic convening, what we do is to bring together the actors that needs to work together to make sure that we are bringing low emission outcomes to bear on all the investments. And then through effective grant making, we approach our grant making through the co-creation process, involving all stakeholders. So in every action that is supported, we are sure that it is going to benefit the stakeholders, including the farmers who do the implementation at the local level. So innovation, we support development of innovation. And I wrote here as a few innovations that are relevant in the rice subsector. Dr. Aidan mentioned a couple of them in the livestock sector. Envisage has been able to produce rice. And just for context also again, the rice that we produce, 90% of them is flooded rice. So that is a key source of the methane that is produced from the rice sector. So can you imagine if we are able to move or shift from flooded rice systems significantly to aerobic rice systems? That automatically cuts the production of methane, which is 15 minutes up? All right. So I just run up now. So we support the development of innovation, and some of the innovations that could be relevant with the discussions we're having here is soil amendments, how they are able to support reduction in methane emissions, and also microbiome modulated methane emissions. Methane emissions is a microbial process basically, and the microbes are in the soil. So how do we understand the dynamics and use that understanding to develop solutions that work across various production regions? So I know Mr. Chairman is on my neck, so I'll just quickly run through. So we develop with stakeholders, we scale for impact, because we can only get the impact when we scale. We cannot get the impact when we are not able to scale. So that is a key aspect of our programming as well. And then we engage. We engage with partners, and as I said, we engage with all the actors on the value chain across the subsectors, livestock and the rice, to make sure that actions are aligned in that way. And then we equip for better data to build stronger systems, and supporting improved reporting, and then projections, improving local capacity to produce data. Data is only relevant when it tells the story from where it is taken from. And so just having the data without understanding what is behind that data doesn't make it actionable. We need actionable data to be able to take the steps needed to reduce greenhouse emissions from the agricultural sector. So I will quickly end here. Thank you very much.
Thank you very much. We have time for 1 or 2 questions and short answers. Yeah. Please, Edward. Benson.
Yeah, so from what we have been working on with the EJP projects, we have come out with some technologies that we still want to do the scale up. And from your approaches, we've realized that you also have a scale up approach and you engage. So how do we work together? Do you have the resources to support us in the scaling up process from what we have generated? Do we approach you to see how we can scale up what we have achieved in the EJP projects for Ghana? Thank you.
Thank you, Dr. Ewa. In the presentation, I also said that we are strategic conveners. Strategic conveners. So if we do not fund you, we can point you to where you can get the fund. No, but the point is that we are very much interested in these discussions, because these are the discussions we look forward to having, and we do the co-creation process to understand better the dynamics of the action that we want to take and how effective it will be where it is going to be implemented. So we are very much open to discussions, and that's why Caleb wanted me to be here, so that we can commence or begin these conversations to identify the opportunities that exist, especially in the Global South, for action to be taken.
Okay, thank you very much for your nice presentation. In terms of the reducing emissions from rice field and then the alternate wetting and drying, I have been wondering if there's a framework to monitor that and then give credit to farmers who do that in our terrain. What is your take on this?
Thank you. It's a very important question. Thank you. So, 2 things. The alternate wet and drying system is perhaps the most popular mitigation strategy across, because it's water management, basically. You drain when the rice plant don't need water, and then you let it in when it needs water. There are challenges to it. The farmer must have control over the water, so that when he needs it, it gets back. So these are some of the scaling challenges. But it is an effective methane mitigation strategy, upwards of 70% emissions reduction. But there also, it does not guarantee productivity gains for the farmer. So the incentives are also low. But this mitigation potential is no doubt, it is there. And they talk about the carbon credit. Yes, it is an incentive that can be used to get farmers to adopt more, because they're going to get returns on the action that they take. But there again, the problem of monitoring comes in. How do you monitor to know the abatement that you are doing? So it is linked together. It is not enough for us to have just the solutions, but we must have the ability to be able to monitor and to track progress of the actions that we are taking. So it is a discussion that is ongoing, and it's a very good opportunity for stakeholders, especially farmers in the Global South, to take advantage of the emerging carbon market to get finance to improve their livelihoods, while also implementing these solutions that cut down on emissions. So the verdict is still out there, and it's a conversation that has to be ongoing to find the best pathways to take advantage of that.
Thank you very much.
Yeah, thank you. That's it.
This is the third speaker.
Okay, good afternoon. I have 10 or 15 minutes.
15 minutes.
15 minutes to present. Okay, that means I'll have to cut things. Yes, I will make it quick. I want to touch on functional carbon capturing materials for regenerative agriculture. And the presentation overview, I'll first talk about carbon capturing materials, how to characterize them, more specifically characterization of carbonaceous nanomaterials, and we look at the MRV frameworks, challenges and future directions. Basically, nature has made it such that carbon can be captured. Trees mainly would do that by taking in carbon dioxide. And we also have the ocean to also take care of that also for us. But then with what is happening or what we are doing to our environment, the uptake from the trees and from the oceans is not adequate. We are doing enough damage to our environment to necessitate intervention by coming up with artificial means that we can uptake or capture carbon dioxide and take it from the environment and store them to make use in agriculture. I'm going to focus on biochar, I'll put it that way, as the first artificial material that can be synthesized to uptake carbon dioxide. Biochar pretty much comes from pyrolysis. That is a thermochemical process where you have your biomass. The biomass can be from wood chips and agricultural residues that you heat in the absence of oxygen. That will give you a char. Here we do it locally, producing charcoal. If you pick the wood and you dig a pit, you cover them, you set it on fire. In the absence of oxygen, you get charcoal. Now, biochar is something like that. Typically, at temperatures from 300 to 700 degrees Celsius, in the absence of oxygen, will give us biochar. And this has the ability to uptake carbon dioxide, capture it, and also store it. What are some of the properties of biochar that makes them desirable? The pores that are introduced in the char are such that they have a high surface area. And these high surface areas are able to trap carbon dioxide. And they do that by what we call adsorption, and the adsorption can be either physisorption. Physisorption is if the adsorption is encouraged or happens because of van der Waals forces. Van der Waals forces are secondary forces that can enable this absorption to happen. Now, if it's not physisorption, then it's also chemisorption. And chemisorption is when you also make use of chemical bonds to uptake or bind onto carbon dioxide to now take it up. Now, biochar is also very stable. So because of the stability, you don't have to worry about the decomposition, basically. Because of time, I will move on quickly. Now, another advantage with biochar also is that there is the ability for doping, nitrogen, oxygen doping. When this doping happens, it increases the uptake of carbon dioxide from the environment about 5 to 10 micromole per gram of the biochar. So biochar, when it's designed this way, is also desirable. Now, I'll also talk about this later because of time. I want to talk about activated carbon. Activated carbon basically is biochar that has been activated at high temperatures from about 400 to 800 degrees Celsius. And this activation can be done by potassium hydroxide or phosphoric acid. And the potassium hydroxide and the phosphoric acid, what it does actually is to increase the porosity within the char or the coal. When we are designing these natural and synthetic materials to uptake carbon dioxide, what we look out for, one, is to make sure that the porosity is high. So how do you increase porosity as much as possible? Because the more porous it is, the higher the ability it will have to bind onto the carbon dioxide, to uptake it, and also to store it. So activation aids in this practice. You can also do it naturally at home if you don't want to do it scientifically. That is, if you have the char or you have a charcoal, you can crush it. And if you crush it and you soak it for about 48 hours in lime, it can also still achieve this activation. And when it's activation, and when it's activated, the uptake of carbon dioxide also dramatically increases. Moving away from charred products, I want us to quickly look at carbonaceous nanomaterials. Because of time, I'm skipping through. Carbonaceous nanomaterials. These carbonaceous nanomaterials include nanoporous carbons, graphene, carbon nanotubes, etc. Now, carbon, as we know, has different allotropes. They exist in different allotropes. These different allotropes will enable carbon to form graphenes, for one. And graphenes are these hexagon-shaped bonds that are put together in a plane. Now, when you have these planes, you can have a number of them, a number of these planes that can be bonded together by van der Waals forces. From graphenes to, you can have what we call buckyballs. Buckyballs can just be just a plane of graphene. But instead of just hexagons, you have about 20 hexagons, and then you have about 12 pentagonal-shaped bonded carbons, which form the C60 buckyball. The C60 buckyball also has the ability to uptake carbon. So now we have graphenes, we have buckyball. The next one is carbon nanotubes. Carbon nanotubes is the cylindrical form of these graphenes. If you have these graphene layers roll up into cylinders, you have carbon nanotubes. Some can be closed-ended, some can be open-ended also. These graphenes or fullerenes, carbon nanotubes, they have a strong and a high ability to uptake carbon dioxide because of the high reactivity of the surface area that they possess, and also they also have high ability to transmit electricity, and then also high thermal conductivity. And also chemically, it's very, very stable. And they have a high ability to capture carbon dioxide and also bond to them and trap them. I want us to look quickly also at their ability to capture carbon in the soils. When you have these carbonaceous nanomaterials integrated into the soils, they capture the carbon dioxide, they keep them in the soil. They also bind together the soil, so when there's rainfall and there's runoff, erosion does not happen because it captures the soil and reduces drastically erosion. When this happens, it helps the farmers. They don't have to spend money on irrigation, for example. They also encourage soil nutrients to also stay within the soil. The loss of soil nutrient also goes down very drastically when these carbon nanoparticles are present within the soil. The last artificial intervention that I will talk about material-wise is composites. Composites of these biochar that I mentioned with carbonaceous nanomaterials. And also we have what we call metal oxide frameworks or covalent oxide frameworks. Now, As I mentioned, what we are trying to achieve is to have a material with a very high porosity. And these high porosities will now enable the material have the ability to capture carbon dioxide. So with the ion frameworks and also the ion frameworks and ion organic frameworks and covalent organic frameworks, what we have is nodes, and these nodes are metal sites. And then the metal sites are linked together by what we call organic compounds. And these organic compounds serve as the struts that hold them together. And then they form pores. And then within these pores, we can capture this carbon dioxide. So metal organic frameworks and covalent organic frameworks also help us achieve that purpose. Now, how do we characterize them? The characterization is also based on the synthesis. For the synthesis methods, we look at pyrolysis and then carbonization. We have hydrothermal carbonization. Carbonization, as I talked about earlier, has to do with if you take the organic compound, you take the wood, and then you heat it in the absence of oxygen, what you have done is pyrolysis. Now, we also have the hydrothermal form, where if you have your organic compound in a liquid state, for example, a sludge, you can also heat it in the absence of oxygen, temperatures to about 500 degrees Celsius. And you also have some of these charts. So the pyrolysis is for the solid one, and the hydrothermal carbonization is also for the liquid one. The next synthesis method is the template-assisted approaches, where you can use silica, pore silica with pores in them that can capture the carbon and then store them. The last one will be the chemical vapor deposition. In chemical vapor deposition, for example, carbon nanotubes. Chemical vapor deposition, if you heat methane, they also presented before me. If you heat methane in a catalytic environment, you can use nickel foil. It's heated at high temperature, then it will break it down, and then it will bond to the substrate and then give you your carbon nanotubes. So that is that for the synthetic methods. Now, how do we characterize these natural materials that can be used to capture carbon and sequester them? The first one, we look at how do you characterize porosity and surface, and also surface analysis. What helps us do that is the BET method that we use for absorption and desorption isotherms to quantify surface area and also determine the pore volume. Also, how do we also determine the density of the porosity within the material that we have synthesized? The next one is determining morphological imaging. We make use of scanning or transmission electron microscopy, SEM and TEM, to visualize the pore hierarchies that we have within the particle size. Typically from about 10 to 100 nanometers, you can make use of the SEM or the TEM to determine that. Now, we also make use of small angle X-ray scattering, nanoscale scattering device also to help determine the sizes. For chemical composition, we use X-ray photoelectron spectroscopy and then Fourier transform infrared spectroscopy to identify functional groups such as carbon oxide bonds. Also, for structural and then thermal probes, we can make use of X-ray diffraction, crystallinity, and thermographimetric analysis to help us also achieve that. Now, I want us to quickly look at some MRV frameworks. For material assessment, we need to evaluate sequestration potential and their durability and life cycle impacts of FCCMs, and also digital monitoring to deploy IoT sensors. These carbon materials I just talked about, they also have high ability to be used as sensors. And if we can leverage that, we can generate some sensors that we can deploy, that we can use to monitor the carbon content of the soil and their storage and their loss, and whether they leach out of the soil or not. We can also make a use of AI modeling to also help us track these flashes in the carbons. Verification metrics establish conversion factors linking application rates to verified carbon storage. Also come up with some policy alignment to ensure scalability and then consistency across diverse agricultural systems. What are some of the challenges and future works we can look at? High cost of production, about $10 to $50 per kilogram of these materials. If you want to also scale it up to the gigaton level, where we can trap gigaton levels of these carbons and then store them, it's expensive. So that is also challenging. And also, it has the ability to degrade in humid environments. So when we have sulfur oxide-rich environments, it can also degrade. So we can make use of AI, for example, to help us in pore optimization. Instead of just leaving it to the natural chemical process to determine the pore that we have, AI can help us to actually quantify the exact porosity we want to achieve for whatever purpose that we want to attain. And also, we need to develop continuous flow synthesis and integrate with renewable energy sources to achieve net zero processes. So by year 2050, if we are able to achieve this, I believe the world will go a long way in solving the carbon problem we are facing now. Thank you very much. Yes.
Thank you, Prince. It's really exciting new developments. I'm here. But with all of these sort of techniques, there is a question of life cycle analysis and the question of scalability. You touched on scalability, but does the cost of actually producing and transporting and delivering these sort of materials on the scale that we need, does that offset the whole carbon costs of which are there in the first place? I mean, I don't know the answer. I'm just asking for your opinion.
Okay, that's a very good question. With the cost of production, as technology advances, technology improves, we'll be able to have ways that we can manufacture these these artificial or synthesize these artificial substances to aid in capturing of the carbon. So with mass production, for example, that can also bring down the cost, and I believe that can offset the cost.
Okay, thank you very much, Dr. Dame. Now, I gathered from your presentation that biochar has the ability to absorb carbon dioxide.
Yes.
Okay, so my question is, do we have other materials or that are capable of absorbing methane also? Methane.
Yes, the biochar can absorb methane. It can absorb methane, but then we want it to absorb carbon dioxide. So I mentioned that if you can dope it, for example, or you can functionalize it by the addition of some imides, and then doping with oxygen, for example, will now have a preferential uptake of the carbon dioxide instead of the methane. So basically, this is focused on the uptake of carbon dioxide rather than methane and other gases that are available, capturing them, storing them in the soil, and also capturing them from the environment to reduce their levels.
Okay, so as in its natural form, like raw biochar will absorb some carbon dioxide, but no methane.
It will absorb some methane in its natural form if you leave it. But then you can synthesize it in such a way that preferentially it will uptake the carbon dioxide instead of the methane.
Okay, by doping, right?
Yes, doping is one of the ways to do it.
Thank you.
Normal charcoal, yes, basically. So that is why when I talk about the actual experimental lab method, I also said that the way we make charcoal here. So a normal charcoal can do that, just that the normal charcoal is not activated. So as I mentioned, you can crush it. If you crush it into the powder form, for example, and then you store it for 48 hours in lime, then it's activated. In that activated form, the uptake of carbon dioxide is really, really high. Yes.
Yes. Yes. But we also have to look at the source of it. We want to solve a problem. We don't create another problem.
Okay.
Because to get the charcoal, we have to cut a tree to get it. So let's take note of that and go to thank you.
Yes. And then can I also add to that? Now, with the current challenge we are having in the country, where there is massive deforestation for galamsey practices and a lot of introduction of heavy metals into the soil, all these synthetic materials I talked about, the biochar, the activated carbon, the carbonaceous nanomaterials, they can also uptake these heavy metals from the soil. So in addition to capturing carbon dioxide, they can also trap these heavy metals. So it's a give and take. It's a give and take, but I believe the advantages here will, overweigh or outweigh what you are presenting.
I want to make a comment on the biochar uptake in heavy metals. I have a PhD student in chemistry who work on rice fields using biochar, and then uptake was good. But one problem with biochar is a reactivation of the heavy metals with time. With time, the heavy metals may be released back when the bonding mechanism is weak. So we need long-term, studies to
research to
exactly
the long-term kicking of the heavy metals when it's trapped. Thank you.
Please. Yeah, it's quite innovative hearing about the biochar and then the liming. I'm just looking at the biochar itself, how the ability to really improve on the pH of the soil. And then the lime itself, we normally use it to correct the acidity. And we are using the biochar also to address the acidity. So when you combine the lime and then the biochar, how does it come out in terms of acidity, correcting the acidity of the soil?
Okay, thank you. Directly, it's not used to correct acidity. The lime comes in when you want to activate it to increase carbon dioxide uptake. That is when you soak it in the lime so that the charcoal is activated and the uptake of carbon dioxide increases. So it's not used directly to deal with the pH or the acidity of the soil.
Thank you very much. I think this is a very motivated topic.
Great.
So I'm calling the last speaker. Yeah, Sinasi. The last. Artificial intelligence.
Hello. And so good evening. Appears it looks like we are all tired. Not yet. And as we talked about a lot of carbon capture things, but we've not talked about how to measure them. Well, things we've done or things that have been done to measure them. So I'm Andrew Salasi Domanu. I'm the Deputy Scientific Director of the Lab. And some of the things we do is to design sensor systems to do wide-scale measurements. So the topic today, AI-powered soil carbon and greenhouse gas monitoring, transforming agriculture through intelligent sensing. So what is the primary challenge? I mean, a lot of time we do measurements manually. We talk about carbon sequestration. We measure everything manually. But what will be the main problem with manual measurements? You have frequency, you have that frequency of measurement you cannot monitor in real time. You also have spatial problems. Manual monitoring, carbon concentration in the first square meter may be different from the next one, or even the next square centimeter, right? But because of cost of human labor, we will need to just take very few samples, right? So the question is, how has AI enabled continuous, scalable, and verifiable monitoring of soil, carbon, and greenhouse gas emission across Africa's landscape? We'll look at what has been done. Basically, what are some of the things we want to measure? I mean, we've talked about it extensively here, so I'll not repeat that. But in measuring from carbon in the soil, or carbon that has been released from the soil, there are various complexity. Even the type of soil, the moisture, temperature, all those things can affect the sensor reading. It's a very complex system. And how do we deal with them, and how has that been done? So we have different types of sensing mechanisms. We have near-field infrared spectroscopy. We also have medium-field infrared spectroscopy. That will give us a much more wider spectrum. We can also use drones. We have multispectral drones, which you can actually map to get different graphs. And to be able to measure these things continuously, you need a working network. So you need an IoT network. So you need a number of sensors that will be connected, that are going to communicate over an established network. So we need to have all those in place. And then we also need to have an intelligence system that can interpret the data. You can do it at the edge, which can process more quickly. I mean, data that's coming in real time, or you can do it in the cloud, which will take. The processing is faster, but it takes a longer time because data will take time to get there, aggregate a lot more data to do processing. Let's see an example in Africa, especially where sensor network has been deployed, what advantages it has given. In Africa, typically when we start talking about technology and and things, we end up going to our friends in the east of Africa, Kenya. So Kenya is one of the first countries in Africa that gives carbon credits. Right, so they have deployed, they have a wide deployment in one of the projects. So they have 50 IoT sensors over 500 acres. Well, if you look at the number of sensors per acre, it looks small. It is expensive, right? But this just 50 in 500 acres. They're able to collect real-time soil data. They're able to look at the greenhouse gas flux, right? Absorption, storage, release, and things like that. They're able to look at it. And then also power. When you have a sensor network, one of the issues is power. And of course, you don't want to use a power source that to contribute to greenhouse gases, right? So solar, they use solar power, and also AI for calibration. Now, what did this do? Right, so they were able to continuously monitor the greenhouse gases at a tenth of the cost of sending humans to go and collect samples, take it to the lab, do measurements, and that cannot be continuous, cannot be real time. If you want to do real time, you have to get people running shifts, maybe a carousel to carry the samples to the lab, right? And that is that's going
be very expensive. Right, so how would this analysis take place? We collect sensor data, and then we can also collect satellite data as well. So we have the sensor data, we have satellite data, we have drone data. We put all together, right? We call it data fusion. We put that together to make meaning out of that, right? Okay, so what are some of the things that are detected? Right? We'll be looking at cover crop, so that's crop cover detection, canopy temperature. We have biomass persistence in a particular area, or changing biomass, and then tillage patterns as well. All those things can be monitored in continuous time. If there's any undesirable change, that can be mitigated early before it becomes a huge problem. So, looking at what some of these MRV practices, so you monitor, you report, and then you can verify or validate. That has been done across Africa, has proven to have very good results. We've collected, over Africa, different projects have collected a lot of data. So over 10,000 plus field measurements, right? And then we've used different AI architectures to look at carbon stock prediction, sequestration rates, greenhouse gas flux forecast. So none that, and on the average across Africa, when we look at soil carbon prediction, we have a very high outstretched score. I think everybody has done some statistics here. So an outstretched score of 0.92, which is what, that means the correlation is very good, right? Meaning that when we collect enough data, I think, was it yesterday or this morning, we are talking about having a lot of data on Senodo that everybody should contribute. Right? Now, to be able to do a lot of prediction, you need a lot of data. So I'll add my voice to Prof. Vincent. So those who have not contributed, right, for these models to work, they need a lot of data. And as we collect a lot of historic data, as all the historic data you have, if you make it available together with everybody's data, we can have better prediction. So let's look at a case study in Zoya. I think if there's any Kenyan there, I hope I have pronounced it correctly. It's called the maize basket of Kenya. So they have deployed this sensor networks, and they give carbon credits. So since they started monitoring, there's been 18% improvement in soil carbon, right, retention. And then there's a 32% reduction in greenhouse gas emission. Okay, thank you. And a whopping $2.4 million in carbon credits. I think that's since 2014, right? it may look small, but this money, otherwise they wouldn't have had, right? So, so they should tell us that once we move in that direction, right, there is also money for the farmers, right? Not just for those providing the solution, the farmers, for everyone. So what we need to move from taking data to action. So we have the full census, we collect data, we do cloud analytics, we can also do edge analytics. Then we use the AI for prediction, we get insights, then the farmers act on the results, right? So that's why we need continuous monitoring. Then when we have this, then we have to democratize access, right? So we say what access should be free, generally. So we have to have in Africa, generally, you know, our official languages are not our mother tongues, right? So if you are developing apps in Ghana, we just don't develop them in English. So as a lab, we also have apps to help the farmers do weather forecast and things like that. We call SIWA, Smart Indigenous Weather App. And the farmers contribute data and they have responses in their local languages, right? So we have to democratize access to this so that farmers can use. And we also keep developing low-cost sensors. Sensors from the global north are quite expensive, and I also use us as a lab. We are also developing sensors for to monitor, to measure, what's the name? Heavy metals in water, right? And I believe once it's ready, some of you can purchase them and help in further development. And to improve research, right, we also need to open source data. So once your Zenodo is fully populated, Zenodo is already open source. So I believe once the data is available, models will be built upon it and we'll have better prediction. And we also talk about partnership. So we are here, we are always open to partnership opportunities as well as Deeper Lab. Okay, so in terms of impact, I mentioned one, that's the deployment reach. There's also financial impact, market growth. I'll skip that and then end because I can see it's almost time. So I'll simply put that the soil can speak and AI helps us listen, interpret, and act. So through this, we transform agriculture, fight climate change, and empower African farmers. Thank you.
Thank you for your interesting presentation. This technology sounds really promising. So you said that monitoring with these sensors is 1/10 of the cost of normal measurements and that you implemented 125 sensors in your case study. But how much does one sensor cost? Because it's probably really pricey to implement this technology.
Okay, so, well, this is not my work. This is from the Kenyan state, I mean the county state. So I'm reporting their work, right? So because of the cost of sensors, I saw my scientific director's hand up. So let me use sensors for heavy metal measurements. Ghana have a problem with Galamsey. So our waters in some of these areas are contaminated. And if we have to do continuous measurement, those sensors are quite expensive. Some are in the thousands of dollars. So what we are trying to do as a lab, but we can't deploy them. They are too expensive. So we are trying to develop sensors less than $100. So that's what we've been doing.
Mine is a general concern about super AI. I read that there are a number of petitions against the development of super AI because of the fear that it will suppress human intelligence. In your field as an AI specialist, what is your take on this?
Well, the AI system is also developed by human intelligence. So there's always evolution. There are pastors who say you should not read your Bible from a tablet. Right? So, as systems are changing, humans evolve. So there may be some basic acts. If, let's say, if you have a washing machine to wash your clothes, it has not made you lazier. It's made you more productive because you use that time to think of other things. Right? So I don't think AI will make us lazier. EJP. That artificial general intelligence. Right? So basically, trying to say it can do everything, think like a human generally. At the end of the day, AI only, it is generative, but it only generates what you have taught it. It puts what you've taught it together to generate. So at the end of the day, I would say, whatever the case, as AI does some, we have spare brain power to use for other things.
Thank you very. Oh, sorry.
Hello. I think it was also in connection to Johannes' question on the MRV for carbon and other stuff. Already, Deeper Lab is liaising with CSIR Crop Research Institute. And with UK, two institutions, Sonar Vision and then the James Houghton Institute, we are coming out with a real-time monitoring device for carbon. Some of these sensors have even been trained to have sensor eyes to be able to see their environment. Because sometimes farmers tell you that if these things are there, it distracts their work, something like plowing and others. So as you are saying, some of these things, the idea is produce them at a cost lower than $100. And then you are able to take some of this data in near real time, sometimes at 30 seconds interval. So you just have to imagine that you are taking this data for about 6 months. You have thousands of data sets to be able to make massive decisions. Yes, it's a very good deal for just $100.
But I think carbon changes will be not happening in 3 seconds.
No, for sure.
Yeah.
Thank you very much, Chair. So you make a point on democratizing access. Thank you. So it's, for me, I think it's contentious in the sense that this is my personal belief, right? I know it goes against what Prof is doing. This is what I've realized. Most funding mandates require us to open up our data sets as Africans, right? So most of our data sets are online, easily accessible. But usually it's difficult for us to come across data sets from US, from Europe. And So the open access mandates, in my view, only applies to us. So in my view, we get a funding, we make it access, and developers outside get access to it. So when you made the point on democratizing access, I just pointed to Prince and said, does this man believe in what he's saying? Because they said data is the new gold, right? And all this gold, you are democratizing it. But we preach open access, but should it be a restricted democracy? Let me say, is it authoritarian democracy or something like that? Is that the way to go? Opening up all our data sets without having any competitive or developmental advantage. And then, of course, because funding requirement says it. Is that the way to go? This is my view. Though.
Basically, the reason why that has become the new norm is that our governments do not fund us. Those funding us are from the global north. And once they are the ones funding, they'll call the tune. So if we really want to develop the way we should, then we should put our monies where our mouth lies. We should. If the government funds, then we can keep the data to ourselves and develop ourselves.
I think it is an end need discussion on that. I remember in 2007 or 2005, I've been leading a European project, a big one, within the framework of 7, and it was one of the discussion about the openness of the data for the industry, and it was a discussion because all public data, because we get funding, our data is not below to us, and even this discussion is still going on at the Indra Institute, at the university, and all. We have to make this data open. Even now, the institution really make that the publication, they pay the fees with publication in a way that open the data. But I told them that at that time, in 2007, 2006, 2007, the European Commission, I told them, okay, it is only one way. That is, we open the data, but the private company, they are not doing the same thing. And I heard the same response. We are not getting money from the government. But they said, because we are paid by the government, we have the obligation to open this data, making it freely for them. That means I think it is not choice. We don't have a choice. And then, of course, as far as we are paid by the government, the data not belong to us. But really, last week, it was the same discussion. We have under pressure. For all evaluation, we have to make clearly that the data is open, that the data is with a code, that the data. But when you talk about the scientists, they have this whole kind of the embargo. They said, oh, I didn't publish, I didn't have time. But they give them a slot time to publish the data. But after a certain time, even some of them, they go retirement without providing this data openly. So I think it is an educational way to behave. And I think that we never change. Even our institution and our government, really, they force us to do these things. But I hope that this kind of collaboration between private sector and government sector must be a flow and an exchange, and otherwise we will stick on that. Yeah.
And also, collecting data is expensive, and the average African business or average African institution cannot afford it. So once we collect small, we aggregate, then we can do something meaningful.
Something, if there is no question, I have to just comment on it. I think with respect to what I'm expecting, maybe I will retire without getting the right answer. So when we're dealing with the ecosystem, agro-ecosystem, is a complex system. when you talk about trying to focus on carbon stocks and then evolution of carbon stock, but carbon stock is not behaving alone. Carbon stock is dependent on the input, output of the vegetation. And this is the question I wanted to answer to the biochar guy, that is producing the biochar, we need the biomass, we need to transform the biomass, and we are lacking the biomass. We don't have the biomass to produce. So we are just turning around, and sometimes we are not answering the right question. The second thing is that this biomass, water, and many other things. I mean, we need several sensors to measure all of these things. But the key question, all of them, as you said, Caleb, that they provide 100,000 of gigabytes of data. What I'm expecting from the artificial intelligence to put this data together, to speed up the process in terms of calculation. Because if you give me this 1,000 data separately fragmented, I can spend 10,000 years also to put them together. Because this is what we say. All of these things has been existed for many. The remote sensing is basically now more than 20 years we have it. And then the carbon measurement, we have it. But linking the two together is a time-consuming. And this is why we need the artificial intelligence to speed up these linkages and articulation of the data to provide a hint on where we are going to move on and what can be provided to maintain a system in a direction, in one direction or another.
Yeah, and one other thing too. We talk about AI, process data. Now, that means we need compute. And compute needs power. And power, then we go back to the same carbon. Right? So the whole cycle, yeah.
Okay. That's the impact of this big server that will store the data in the coming year is basically as equal to a city of New York, what is consumed in one night. It's huge, really it's huge. And this data this energy should be coming from somewhere. Anyway, we have to keep an eyes positive and not.
All right, thank you.
Yes, please. I don't know.
Yeah, hello. Yeah, so I'm a mathematics major person. Like, I don't really know much about the soil stuff and all those stuff. But I want to ask, like, in all this, what is the future of mathematics and which particular IT skills come in handy?
Future of mathematics?
Yeah, in this.
It's like asking me what's the future of a farmer when it comes to food.
Okay.
Yeah, because basically these algorithms were developed by mathematicians and computer scientists. So really, maths is life. So I'm talking as an engineer, right? Yeah, mathematics is life. So really, there is no problem at all.
So which particular mathematics skills or IT skill?
Would you need, okay, depends. Is that you are looking at moving a particular field, that's a machine learning direction. Okay. For that, basically machine learning, statistics, linear algebra, those are the probability stats, linear algebra, that direction. For programming, any programming skill, it's just that now predominantly it is Python, right? But you can use MATLAB, you can decide to even use C, right? Which might even compile faster, which will be faster. So now it is predominantly Python. There's Julia, but I don't think language is a problem. In the next 5 years, maybe something else will come and Python will be pushed to the background, right? So for that is not an issue. Okay.
Hello. Melissa, are you online? Yeah. Hello. Yeah, it was a really long day. She started almost around 3 a.m. in Brazil. So,
1.5.
it's what?
Now
Yes, it's still
1.5.
there, but she has started long. Yeah. Okay, we are done for the day. It was an interesting day. Tomorrow we're going to meet a lot of the next generation, that is postgraduate students, mainly masters and PhDs. Okay, Melissa, can you give us a closing remark for the day's activity?
For today?
I know today, like a closing remark for the end of the day.
Wow. I took some notes from all the day. I think I'm very happy for those discussions. I took some notes for my questions here. I took some for one for Caleb about the integrated crop livestock systems. And I'm very interesting about that. Sorry, let me close my window. Very interested about those information shared by Dr. Coffey about the global methane hub, because here we are. I'm an employee from Embrapa Rice and Beans. So it's very interesting, those questions, those information that he shared with us. So I think we, as a project, we could discuss and work together, because I think we have lots of information and lots of things that we could share. And yes, this meeting today, I was a very high level of discussions. Here, according to our program, we should put in line what we should go through about our project extension. But I think we already discussed this.
Yeah.
I send again a chart. Here I would like to have Abad help here, because I have lots of information, but I can't put this in a way because my mind is full of notes here. Yes, I have so much so much ways to think, to go about our project. I really I wasn't expecting this kind of information, so strong information from so many huge researchers. So I'm very happy with this. Yay! So Abad, what do you think about our day today? I would like to hear you, because I'm here of the other side of the globe, and you are together with our team, in Ghana.
Yeah.
So could you share with me and share with the team about what we saw today?
Yeah, of course. Yes, of course. From what we heard today, this was a very rich day. It was very how we call, lots of information, diversify information, touching all level. And the question is now is how we are going to digest all of this information, how we can put them together, which one are more responsive to the project and the objective of the project. I think basically most of them. So now the question based from what we hear today and then how we are going to move on in the next coming days and until the end of the project. And I think it's very important from what we heard, and especially I love this the afternoon about the regenerative agriculture, but also this morning with the overview for each work package and where we are. Of course, it's still some work to be done. The project is still ongoing. It is not finished. We know now who is doing good, who need to improve, and then what are the gaps that need to be fulfilled. And of course, yeah. But it was a very exciting. I didn't expect this high-level discussion we have had, and thank you for the organization. For the African colleagues, really, we are only here 3-4 people, but the majority of you is making this workshop really successful, and thank you all of you. This is what so this is what I can say, Melissa. I don't know if you are happy with that, but anyway, yeah.
Yes, I'm without words.
The only things, I think if it is possible to have this, because if we want to, I think we have to do it, at least if we can have a copy of this presentation. It's very important for us to make a kind of small report, at least 1 or 2 pages to highlight what's going, because we need some, how we call that, footprint of this meeting, not that just discussion with the implication and then what will be the next step to follow. So it's, if you can have this presentation all together in a PDF file for every day, and I take some of the notes, but will be very helpful for us and for Melissa and for the coordination level, but also for you also to make a track, and then can be also on the website of the project that people can say. We just discussed a few minutes later about the extent of openness of the data that make this data democratically open in a way that everybody can get the data, knows what's going on in this project. For the founders also, it's always good.
Thank you, Abad. Luckily for us, we have all the presentations, so I'm going to put it into a Google Drive so that everybody can access.
Yes, great. I asked to Caleb about a list of colleagues that are in the room. This is very important later too. It will be very important later to see how many people was participating, because from here we have the people who was in the virtual meeting. And it's very, very important. About the event until now, again, I'm very, very happy. Yes, for my huge thank you for the team from colleagues from KNUST and partners from Namibia, from South Africa, and for students. You are, as our colleague said, you are the next generation. So I'm very proud to have you with us today and the next days. So I think I'm not able to have a so huge event, to produce so huge event. So I'm really count of you to do this. All the team do this by yourself. So in English, it's very hard to express my my gratitude about this is happening. So thank you very much for this. So I'm very happy for this. Thank you.
Melissa.
Yes.
Yeah. So just because we have a section now after for the extension, the project extension. So I got a message from our funding agency. They said, okay, there is no problem. We have to do it online with the letter together. But they are really, I think that we have been struggling last year about the report. So far, we didn't provide the report. There is a control on the report, and they are really, the red lights are open, and they need the report. You remember last year, we didn't get this for Puerto Rico, and now really they sent me, I received 2 messages this afternoon that ask that we need this report to be downloaded on the funding agency. I don't know with the other country, but in France they need it.
No, in fact, in Brazil we have to have these two. I confess I forgot to ask after, because at that time I was trying and hard.
Yeah, I remember. It was
hard
in
way,
this.
but
yeah, they have had some problem with the internet. Yeah.
Yes,
should I send an email to Ulrika, or should you send an email, or how it work? Or at least you have basically the version that you submit.
Yes, I submit by platform, and I was trying to recover this because they received it, but they didn't. I think they couldn't allow us to proceed the download.
We need a copy of it.
Yes, I will try to obtain this. Let me take note, because otherwise I can forget again.
So please ask Ulrika, because the funding agency, they ask. I know that this is on the project management, but the funding agency, they need it, especially in France. They need it. It's still now.
Okay.
It's still
We have the information, but we don't have it in the platform, in the EJP form. We have all the information, but we don't have this in his paper.
The question if we can download it.
I'll try to.
Yeah. Okay. Thank you very much. I send you an email on this.
Okay, we are done for the day. Thank you, all guests and all participants, for making the day successful. Thank you also for the online participants to stay till this time. I know it's very, very boring to be online for more than 5 hours, but you've been able to exceed about 8 hours so far. Thank you for your patience, and thank you to everybody around.
Thank you, guys. Thank you very much.
The meeting is adjourned till tomorrow at 9 o'clock.
Okay. See you tomorrow. Bye-bye.